In animal development, cell fate and potency are progressively restricted from totipotent to unipotent until a cell turns into a terminally differentiated, meaning that it could only form a singular cell type. Stem cells are different because they never reach the end of their development. As each choice in the developmental hierarchy limits what will happen to a cell, we say that the cell is committed to a certain fate. Animals show their commitment in stages. Specification, induction, and competency. Embryo interacts alongside its environment, as well as its developmental trajectory could be guided by information from its surroundings. On a relatively small scale, an embryonic cell's environment is made up of the tissues around it in the embryo. The fate of a cell, like whether it will become part of the skin or the lens, often depends on how it interacts with other parts of its immediate ecosystem. So, a second experimental embryology research program looks at how interactions between embryonic cells make the embryo. Specification and determination. These steps of specification and determination are involved in planning the cellular identity. Once specification has taken place, a cell will be committed to differentiate down a specific pathway if left in its normal environment. The steps of commitment could be classified into two stages. The initial stage serves as a labile phase termed as specification. The fate of a cell or otherwise a tissue is marked to be specified whenever it is capable of autonomous differentiation while placed within a neutral environment. Like a petri dish test tube. In this stage, the Kami-T net can be reversed. 5. Development biology. Basics of development. Potency and commitment. A major walkthrough. Cellular differentiation serves as a key step by which the cells typically become structurally and functionally specialized, which allows the formation of distinct types of cells. For instance, the nervous system is, in turn, made up of various types of neurons and glial cells. The structure alongside functional specificity of a cell in fact depends upon the proteins that are being synthesized. In the recent past, the emerging evidence highlights that regulation of non-coding RNA makes up multiple developmental steps alongside cellular differentiation. With few outliers, all of the cells within a given organism follow genomic equivalence, implying that the genetic information is the same in all cells. Differentiated cells are genetically identical yet express different genes. Thus, the steps of differentiation involve the control and maintenance of differential gene expression. Fate and commitment. The number of types of variety of cells within the embryo elevates as the development proceeds. Cellular fate marks the range of cellular types that a particular cell could give rise to in the course of normal development. Cellular potency marks the total repertoire of cellular types a particular cell could give rise to in all possible environments, suggesting that a cell could differentiate by an abnormally weight if all it is experimentally being grafted into an ectopic region. The cellular potency serves as an intrinsic property that could be greater than otherwise equivalent to its fate. The fate of a cell depends on its potency, as well as autonomous specification generates a pattern of development termed as mosaic development, as the embryo looks as though it is constructed like a mosaic tile with self-differentiating independent parts. Invertebrate embryos, with special reference to annelids, tunicates, and mollusks, exhibit autonomous specification. Within these embryos, certain proteins, otherwise messenger RNAs, form the morphogenic determinants. These are positioned within various regions of the egg cytoplasm and are proportioned to the different cells when the embryo divides. Such morphogenic determinants only specify the nature of cell type. The next phase of commitment is termed as determination. A cell or tissue is determined when it has the capability to differentiate autonomously while being placed into any other region of the embryo. This ability to differentiate is for its original fate irrespective of the surrounding conditions reveals that the commitment has become irreversible. Autonomous specification. There are three types of commitments. The key type is termed as autonomous specification. As for this specification, if a specific elastomere is taken away from an early embryo, the isolated region would be able to generate the same set of cells that it would have produced even if it could have been a part of the embryo. Figure 5.1. Autonomous specification. AC. Trochoblast. Facilitated cells differentiation in Mollusc patella. A. The 16 cell stage is observed from the side. The presumptive trochoblast cells are being shaded. B. 48 cell stage. C. Facilitated larva stage, as observed from the animal pole. D. G. Patella undergoing trochoblast differentiation from a 16 cell stage and cultured in vitro. E. F. First and second divisions in culture. G. Facilitated products of F. With isolation, the cells undergo division and ciliating at the right time after Wilson 1904. Figure 5.2. Autonomous specification in the early tunicate embryo. While the 4 blastomere pairs belonging to the 8-cell embryo become dissociated, each of them forms structures that it will form if it remains within the embryo. The tunicate fate map depicts that the left and right sides generate identical cellular lineages after rubber bearing in 1946. Figure 5.3 is equal nest race in the progeny of the muscle lineage blastomeres. B4.1 isolated from a tunicate embryo at the 8-cell stage. The B4.1 blastomere pair progeny C, yet not within the 6/8 of the remaining embryo D. When incubated for a specific time, it forms a larva. From Whitaker 1977. Photographs courtesy of J.R. Whitaker. A. Isolation procedure diagram. B. Acetylcholinesterace nest race localization within the tail muscles of an intact tunicate larva. The enzyme's presence is represented by the dark staining. The dark staining is also noted within. Figure 5.4 on tunicate eggs. Microsurgery pushes the yellow crescent cytoplasm of the B4.1 blastomeres that are muscle forming to make an entry into the B4.2, which is the producer of skin, as well as nerve blastomere pair. The push of B4.1 blastomeres with a blast needle creates cleavage furrow regression. The furrow will in turn reform at vegetal positions where the cells are being cut alongside by way of needle. The new furrow thereby segregates the cells by way that the B4.2 blastomeres will receive certain muscle forming yellow crescent B4.1 cytoplasm. Such altered B4.2 cells will generate muscle cells in a normal ectodermal progeny. Whitaker 1983. Figure 5.5 conditional specification. A. Cell specification depends upon its position within the embryo. Its fate is being determined by way of interactions with the neighboring cells. B. If the cells are being removed from that of the embryo, the remaining cells could regulate and compensate for the sake of the missing part. Germplasm theory. The research that led to finding conditional specification started with testing a theory that said there was nothing like it. In 1883, August Weismann came up with proposed first model of cellular specification, termed as the germplasm theory. With the available information about fertilization at that period, Weismann claimed that the sperm and egg provides equivalent contributions towards chromosomes, both in terms of quantitative as well as qualitative, with respect to the new organism. Furthermore, it was postulated that chromosomes carry the inheritance potentials of new organisms. Also, not all of the determinants localized upon the chromosomes enter the cells within the embryo. Rather than dividing equally, the chromosomes are expected to undergo division in such a way that various chromosomal determinants will enter various cells. The fertilized egg would carry the wholesome complement of determinants, while few somatic cells would have the possession of the blood-forming determinants, whereas others would have the determinants for muscle formation. Within the nucleus of cells that are destined to form gametes, namely the germ cells, these contain all types of determinants thought to be retained. The other cell nuclei will have only a subset of actual determinant types. Figure 5.6 Weismann's inheritance theory. The germ cell produces somatic cells that differentiate to form the body cells and new germ cells. Weismann hypothetically proposed that the germ cells only contain maximal inherited determinants. These somatic cells were first believed to contain a determinant subset. The determinants types that are found in the nucleus will in turn determine the cellular type. Wilson 1896. While proposing this model, Weismann put forward a hypothesis about development that can be tested instantly. With reference to the frog embryos, fate map, Weismann concluded that while the first cleavage division segregated the embryo's futuristic right half from the left half, he insisted that there will be a segregation of determinants of right from that of left, which in turn led to blastomeres. The subsequent testing of this proposed hypothesis pioneered 3 of the 4 vital techniques used up in experimental embryology. The defect experiment wherein one destroys a portion of the embryo and then observes the further growth of the embryo that was experimentally impaired. The isolation experiment wherein one removes a portion of the embryo and then observes the further growth of the partial embryo and the part that was isolated. The recombination experiment is where we observe the further growth of the embryo even after swapping an original part with a derived part originating from a region that is different. The transplantation experiment, wherein one portion of the embryo is replaced by a portion from a different embryo. This fourth technique was utilized by certain scientists whilst they firstly constructed the early embryo's fate maps. Figure 5.7 Roos depicted mosaic development. Destruction yet not removing one of cells within a 2-cell frog embryo illustrates the development of only embryos one half. Figure 5.8 Trias illustration about regulated development. A 4-cell intact embryo C urchin generates a pluvius larva normally. B when a 4-cell embryo is removed from that of the fertilization envelope, and if this 4-cell are isolated, each cell can form a small yet normal pluvius larva. The 4 larvae that are developing in this way are perhaps not identical, in spite of their capacity to generate all of the necessary cellular types. These variations are also noticed amongst adult sea urchins that are formed in this manner. Marcus 1979. Figure 5.9 Dreas pressure plate experiment for the sake of altering the nuclei distribution. A routine cleavage happens within 8 to 16 cell sea urchin embryos, observed from animal pole and from the side. B quite abnormal planes of cleavage are creates under pressure, as observed from the animal pole and from the side. Huxley and Degger 1934. Investigator organism type of experiment conclusion. Interpretation concerning potency and fate. Root 1888 frog Rana fusca defect mosaic development. Autonomous. Prospective potency is equivalent to prospective fate. Dries 1892. Sea urchin echinus microtuberculatus isolation regulated development. Conditional. Prospective. Potency remains higher relative to prospective fate. Dries 1893. Sea urchin echinus and paracentrotus recombination regulated development. Conditional. Prospective. Potency is elevated as compared to prospective fate. Table 5.1 Experimental procedures and results of ruined Dries. Gametogenesis, fertilization, and early development. Introduction. In this subsection, we will see in detail about the gametogenesis step, both spermatogenesis and oogenesis alongside fertilization. Under fertilization, we will see key points like external fertilization in sea urchins, internal fertilization in mammals, and human fertilization as well. From this chapter earlier, several previous years questions have come from fertilization. The testis is routinely positioned inside a pouch like that of a scrotum, which typically holds them external to the main body structure. This favors spermatogenesis, which requires 2 - 3 degrees Celsius lower temperature relative to normal body temperature. Spermatogenesis happens in 2 phases: spermatocytogenesis and spermiogenesis. Spermatocytogenesis. Spermatocytogenesis, the male equivalent of gametocytogenesis, is the process by which stem cells replace themselves and generate a whole new population of cells that will develop into functional sperm. Spermiogenesis. Within the seminiferous tubules, adjacent to the inner tubule wall, the stem cells divide in a centripetal direction, beginning from the outer side, as well as proceeding towards the central region termed lumen. Amongst humans, testes generate approximately 200 to 300 million sperms daily, out of which only 100 million become viable, and the complete spermatogenesis takes place in 74 days. Spermatogenesis steps begin in seminiferous tubules. The seminiferous tubules have 2 types of cells: Sertoli cell and germinal cell spermatogonia. The arrangement of Sertoli cells in the seminiferous tubules divides the seminiferous tubules into 2 compartments: white circle abluminal compartment, white circle basal compartment. Spermatogenesis steps, figure 5.10. In the germ stem cells term type of spermatogonia. Type of spermatogonia lie in the basal compartment of seminiferous tubules. Capacitation, polyspermy prevention, and human fertilization. So, this chapter is critical for the sake of students. Gametogenesis, the gamete production. It is indeed a biological step where haploid gametes mature from diploid, otherwise haploid precursor cells subsequent to cell division. Diploid germ cells undergo meiotic division that leads to generation of various gametes. Gametogenesis happens in gonads, male testis, female ovary, that lead to generation of sperm and ova, respectively. The origin of gametes is marked from germ cells of embryo under development, and they remain unique from the remaining somatic cells. Germ cells proliferate in gonads by way of mitosis, whilst gamete formation occurs by meiosis. Gametes development in the embryonic stage remains the same in both cases of males and females till they become gametogonia. Such gametangia are termed as the successor of primordial germ cells, PGCs. These PGCs originate from endoderm, yet remember, the gonads have mesodermal origin. PGCs are well known to be present in the posterior epiblast that in due time migrate to endoderm. These PGCs undergo migration through the gut to the genital ridges. Spermatogenesis. The step of spermatogenesis happens within the seminiferous epithelium within testis. This seminiferous epithelium is in turn made up of Sertoli cells, somatic cells with germ cells. Figure 5.10 Spermatogenesis, the sperm formation. Upon cytogenesis, these cells move in a spiral manner towards the tubules. The initial spermatocytogenesis exhibits divisions where the nucleus actually divides, yet the cytoplasm of daughter cells still remains connected by way of thin cytoplasmic connections. The succeeding type B spermatogonia in turn divide neotically by producing diploid primary spermatocyte. The primary spermatocyte divides neotically to give else to haploid secondary spermatocyte. The meiosis II occurs in 2 degrees spermatocyte and leads to the generation of spermatids. Spermatogenesis. It is the key transformation of a spermatid to a sperm-like matured structure. It is a transformation step where actually the spermatids undergo differentiation to form male germ cells. The steps involved in spermatogenesis are listed in table 5.2 below. Steps involved in spermatogenesis. Nuclear condensation arginine-rich protein expression replaces nucleosome making further DNA compaction. Acrosome formation Golgi apparatus vesicles merge and enriched alongside lysosomes containing enzymes like hyaluronidase and proteases. Flagellum formation centrioles of the cell undergo elongation to form the tail of the sperm. One among the centrioles will grow with 9 double microtubules in the periphery and in the center 2 singlets are observed. It has 3 parts to name: the neck, the midpiece, and the tail. The midpiece is being wrapped by the mitochondrial group and the tail with the axoneme in 9+2 manner. Cytoplasm reduction. The unwanted organelles with cytoplasm is phagocytized by way of Sertoli cells and disposed of in the tubular lumens. Formation of tail with centrioles elongate to generate the sperm tail. An intermediate structure termed as a manchet in turn helps in the process of elongation. While this phase is going on, the spermatozoa which is developing will orient themselves in a manner that their tails will point towards the lumen center, while it will point away from that of the epithelium. Table 5.2 steps involved in spermatogenesis. Time frame. For humans, the total spermatogenesis takes place for the sake of 74 days, inclusive of the transport on the ductal system. It takes 3 months. Testes routinely form 200 to 300 million sperms per day. Sertoli cell. A Sertoli cell serves as nurse, cell providing nourishment towards sperm development. FSH receptors localized on the Sertoli cell. FSH is being released by the pituitary gland and stimulates the proliferation of the Sertoli cell. This serves as a key point explaining about the testis enlargement in puberty. Sertoli cells serve as phagocytes, taking up the residual cytoplasm in the event of spermatogenesis. The substances secreted by Sertoli cells are listed in table 5.3. Secreted substance function. Anti-Müllerian hormone AMH. AMH regresses the Müllerian duct in the male. Inhibin and activin secreted post-puberty. The inhibin duly inhibits the FSH secretion, while the activins activate the FSH secretion. Androgen binding protein testosterone binding globulin binds alongside the testosterone and increases concentration of testosterone. Estradiol Sertoli cells generate aromatase, which in turn converts testosterone to form 17-beta estradiol that directs spermatogenesis. Glial cell line derived neurotrophic factor GDNF. The capacity of self-renewal in spermatogonia is assured by way of GDNF. The X-related molecule ERM transcription factor. These molecules stabilize the spermatogonial stem cell in the adult testis blood testis barrier. Table 5.3. The following compounds are secreted by Sertoli cells. Sertoli cells are required for sake of male sexual development. In due course of male development, the gene SRY localized on Y chromosome in turn activates SOX9. In turn, SOX9 activates fibroblast growth factor 9 FGF9. The proliferation and differentiation of the Sertoli cell are mainly activated by FGF9. The FGF9 lac tends to induce female development. Once fully differentiated, the Sertoli cell is unable to proliferate. Thereby, if spermatogenesis has commenced, further Sertoli cells are not generated. Oogenesis, the ova formation. Oogenesis happens in the ovary. Primordial germ cells PGCs give rise to eggs, otherwise ova. Oocytogenesis. Primordial germ cells PGCs originate from posterior proximal epiblast junction of extraembryonic ectoderm, allen to its primitive streak in epiblast by way of BMP signal supplied by the extraembryonic ectoderm. Direct migration of PGCs from epiblast to hindgut endoderm takes place during 8th day of fertilization. TR protein, an RNA binding protein halts the PGCs apoptosis and aids in the passage of PGCs. Migration is done with the help of fibronectin and integrin protein. Stem cell factor SCF, c-KIT protein, and ligand KL are essential for sake of PGC survival and motility. Within the female embryo, the gonadal ridge serves as the site of PGC localization. It is a location for sake of maintaining their number by way of proliferation. Besides, they get covered by the colonic epithelium. Proliferating PGC establishes within the developing ovary that differentiates into oogonia. Oogonia are accountable for sake of ova formation. Within the female embryo, during the fetal development, PGCs undergoes meiotic division to form oogonia within the 4th to 8th week of the pregnancy and reach up to 70, 7 million by the end of 5th month. Up to 1 to 2 million oogonia get declines after 7th month, otherwise shortly after birth. Thus only 5 million oogonia the zona pellucida. Corona radiata is attached to cumulus and oocyte through gap junction, forming an electrophysiological syncytium, and corona radiata maintains a close contact with oocyte through cytoplasmic extensions across the zona pellucida. The theca interna serves as major source of androgen hormone. It is vital for sake of the final stage of development of the Graafian follicle. Follicles are classified as primordial follicles, preantral follicles, primary and secondary follicles, antral, and preovulatory follicles. Fertilization. Fertilization is inclusive of all the steps that occur subsequent to the gametes that lead the gonads up to the nuclear and the zygote activation. The perfect fusion linking the sperm and egg is amphonixis syngony. Fertilization indicates the events which activate the sperm and egg. 3 forms of fertilization are 1. External fertilization noted in sea urchins. 2. Internal fertilization noted in mammals. 3. Double fertilization noted in angiosperm plants. Crucial stages in fertilization. 1. Contact and recognition amongst the sperm and egg. Routinely, this phase ensures that both the sperm and egg belong to exactly the same species. 2. Sperm entry regulation. By nature, one of the sperm nuclei only could fuse to the egg nucleus that ensures polysomy prevention. 3. Genetic material fusion. Nuclei of sperm and egg fuse with each other. 4. Egg metabolism activation to commence further development. External fertilization. Sea urchins are a classic example of external fertilization. They release the gametes externally into the environment as they follow external mode of fertilization. Environment might represent a small tide pool else large. Left, which stop dividing and now turn primordial follicle, and the primordial follicle get differentiated to form primary 1 degree oocyte enter in the first meiotic division. Oogonia. Oocytes formed by the oogonia have the nature of stem cells. Oogonia contains follicle termed as primordial follicle. Primordial follicle differentiates to form primary follicle, otherwise primary oocyte, which divides by meiosis either after the completion of oocytogenesis before otherwise shortly after birth. After puberty, primary oocytes express growth by receiving the FSH. Ovary. In mammals, female gonad is termed as an ovary. Ovary serves as endocrine gland that releases steroids, key to female secondary sexual characteristics, and indeed supports the pregnancy. Germinal epithelium is localized as the outermost ovary cover. Located beneath them is another layer termed as tunic albuginea, made up of connective tissue. The region under the tunic albuginea is termed as ovarian cortex that has elastic fiber, fibroblasts, and collagen, while all of them surround the ovarian follicles. 3 major events which took place during the early stages of gonadogenesis responsible for sake of the formation of a functional ovary, formation of follicles, differentiation of steroid-producing cells, and initiation of meiosis. Follicle. An ovarian follicle has different cell types serving as a complex unit. The mature preovulatory follicle has granulosa cells layers. Granulosa cells surround the oocyte while the granulosa cells are in turn surrounded by the theca cell layer. Granulosa cells have specialized subpopulations of cells that diverge in a discrete ion on receptors and steroidogenic characteristics termed as cumulus cells, corona radiata, neural, and antral granulosa cells. The cumulus cells surround the oocyte and nourish them, forming motion. These places are always being shared alongside other species too, which shed their own gametes at the identical time. Figure 5.11 Summary of events leading to fusion of egg and sperm. The summary of events leading to fusion of egg and sperm is illustrated in Figure 5.2. The step 1 indicates the chemical traction of sperm to egg, while step 2 indicates contact with egg jelly, and step 3 triggers the acrosomal reaction, and step 4 makes the sperm to adhere to the vitally envelope and lyses the whole. Step 5 makes sperm to adhere to egg membrane. Now the sperm from nucleus enters egg cytoplasm. Species-specific attraction of sperm was reported in various species. For sake of instance, mollusks, cnidarians, echinoderms, urochordates, and amphibians. In a variety of species, sperms are being attracted towards eggs of respective species by way of chemotaxis that follows a chemical gradient that is being secreted by way of the egg. Such oocytes might control not only the sperm type they attract, also it represents the time by which it attracts them, and thereby they release the chemotactic factors once they mature. Amongst sea urchins, the chemotactic factors serve as smaller peptides termed as the sperm-activating peptides, SAPs, which diffuse gradient from the egg jelly to the neighboring seawater. Classical example of SAP is RESACT, which is a 14 amino acid peptide being isolated from that of the egg jelly from the artesia punctulate sea urchin. There is a profound outcome even at very low concentrations in way that the binding of single RESACT moiety is optimal to provide the directive route for sake of the sperm to swim towards the concentration gradient set up till they reach the egg. Sperm with the egg jelly. Protein assimilating enzymes are being released from acrosomes that digest a path throughout the jelly coat towards the egg. After the sperm reaches the surface of the egg, the acrosomal steps that stick to the vitline envelope, which tethers the sperm to egg. It is probable that hefty complexes of these acrosomal protein assimilating enzymes digest the vitline envelope at time of attachment and progress towards the cellular membrane of the egg. Acrosome reaction. Another major interaction among sperm and egg jelly marks the acrosome reaction. Major marine invertebrates has 2 workings as given in Figure 5.12. White circle fusion of the acrosomal vesicle alongside the sperm cell membrane. It represents an exocytosis, which consequences to release the acrosomal vesicle contents. White circle extension of cellular protrusion is. Turn the acrosomal steps. The acrosomal reaction alongside the sea urchins is triggered by way of contact of. Figure 5.12 Acrosomal reaction and exposure of bindin. Figure 5.13 Species-specific induction of acrosome reaction. Further diffusion of sperm egg causes the actin polymerization within the egg to create a fertilization cone. Actin forms a connection that in turn widens the cytoplasmic ridge amongst the egg and sperm. The sperm's centriole, nucleus, and the tail travel through this ridge. Fusion serves as an active step, mediated by way of explicit fusogenic proteins. Among sea urchins, bindin serves the second role of fusogenic protein. Apart from egg recognition, bindin has long stretch of hydrophobic amino acids adjacent to its amino terminus, which fuses the phospholipid vesicles. Polyspermy prevention. 1 sperm for sake of 1 egg. In sea urchins, like the majority of animal models studied, any of the sperm that makes an entry into the egg could provide the egg with a centriole and a haploid nucleus. In normal monospermy, in which only 1 sperm enters the egg, a haploid sperm nucleus and egg nucleus that is haploid in nature combine to generate the fertilized egg nucleus, which is diploid remarked as zygote, thereby restoring the appropriate chromosome number for sake of the species. During cleavage, the sperm-supplied centriole splits in half to form the meiotic spindle's 2 poles. Most organisms cannot survive the introduction of multiple sperm called polyspermy. When 2 sperm fertilize a single egg in a sea urchin, the resulting nucleus is triploid, with 3 copies of each chromosome rather than the usual 2. Worse, since each sperm centriole divides to form the 2 poles of a meiotic apparatus, instead of a bipolar meiotic spindle separating the chromosomes into 2 cells, the triploid chromosomes might be divided into as many as 4 cells. After Alves et al., 1997, J. Biochem 272, 6965-6971. Figure 5.14 Species-specific binding of acrosomal steps to egg surface in sea urchin, C. G. Glab and B. D. Gapier, 1977. Nature 267, 836-838. Eggs extracellular coat recognition. Sea urchin sperms encounter the egg's jelly coat components, which give the primary set of events that are leading to species-specific recognition by progress of sperm attraction, sperm activation, and acrosomal reaction. Yet, additional binding events that are species-specific must take place when the sperm has completely penetrated the egg jelly and its acrosomal steps contacts the surface of the egg. The acrosomal protein that mediates this recognition among sea urchins is a 30,500 insoluble protein termed as bindin. Acrosome containing bindin of strong bile centroids was capable of binding only to the jelly eggs of the same species. Further, the sperm bindin is also species-specific. Fusion of the egg and sperm cell membranes. After the sperm has reached the egg and completed the acrosome reaction, the sperm cell membrane fuses with the egg. This change is caused by a small influx of sodium ions into the egg. Sperm could fuse with membranes that have -70 millivolts as a resting potential. They could not fuse with membranes that have a positive resting potential, and this ensures that there is no further binding of sperm with the egg. It is indeed not familiar that the elevated sodium permeability is owing to the contact of the first sperm or else due to the fusion of the first sperm with the egg. The due significance of sodium ions, as well as the resting potential change, was illustrated by Lorinda Jaffe and colleagues. They reported that polyspermy could be induced if sea urchin eggs are being externally supplied with an electric current that indeed maintains negative membrane potential. In contrast, it would be possible to prevent fertilization by keeping the membrane potential of eggs at a constant positive value using artificial means. The sperms have a component that is voltage sensitive, probably a positively charged fusogenic protein, as well as the insertion of this voltage sensitive component into the egg plasma membrane across the membrane. Electric charge could be regulated. This electric block towards polyspermy prevention is quite expressive in frogs, yet probably not much noticed among mammals. Polyspermy is of 2 types. Pathological polyspermy. All the nucleus of sperm fuses with nucleus of egg. Egg degenerates and embryo do not develop. Physiological polyspermy. Nucleus of only 1 sperm fuses with nucleus of the egg and all the other sperms die in cytoplasm of egg. The dead sperms in cytoplasm of egg are known as merocyte. The fast block to polyspermy. The polyspermy fast block is attained by way of alteration of the egg plasma membrane electric potential. This membrane generates a selective barrier which links the egg cytoplasm and the outside environment. Also, the egg's ionic concentration differs significantly as compared with its surroundings. This difference in concentration is highly vital for the sake of sodium and potassium ions. The constant difference in charge across the egg plasma membrane can be measured if an electrode is inserted into an egg and another is placed outside of it. This membrane potential in resting state is routinely about 70 millivolts, which is expressed as -70 millivolts if the cell inside is negatively charged with respect to the exterior. Within 1 to 3 seconds post binding of initial sperm, there is a shift towards positive membrane potential, which is near plus 20 millivolts. Figure 5.15 change in charge 1 sperm added from minus 70 MeV to plus 20 millivolts. Now plus percentage of. Since the sea urchin egg's membrane potential only stays positive for about a minute, the fast barrier to polyspermy is only temporary. This short potential shift is indeed not sufficient enough to prevent polyspermy that could still happen if the sperm is able to bind the vitally envelope and is not, however, discarded. This removal is being achieved by way of the cortical granule reaction, which is indeed a slower, mechanical block towards polyspermy which remains active for maximum a minute post the first sperm egg attachment, which happens successfully. Directly beside the egg plasma membrane of sea urchin, there are approximately 15,000 cortical granules, each of them are approximately 1 micrometer in diameter. Upon entry of the sperm, such cortical granules will fuse with the egg plasma membrane. They will release their components into the space that links the plasma membrane, as well as the fibrous native envelope proteins of vitelline. Numerous proteins are being secreted by way of cortical granule exocytosis. Figure 5.16 role of inositol phosphates in the release of calcium from Turn hardens the fertilization envelope by way of cross-linking the tyrosine residues localized on adjacent proteins. As it forms, bound sperm are released from the envelope. This step starts about 20 seconds post-sperm attachment, while the completion will take place by the end of the first minute of fertilization. By last, a fourth cortical granule protein, hyaline, triggers a coating around the egg. The egg in turn extends the elongated microvilli, which have tips that are being attached to this hyaline layer. This layer in turn offers support for the sake of the blastomeres during cleavage. The first are proteases. Such enzymes aid to dissolve the protein which connects the vitelline envelope proteins with the cellular membrane. Also, they will clip off the attached spin receptor along with any of the sperm attached to it. Mucopolysaccharides secreted by way of the cortical granules can in turn generate an osmotic gradient that makes water to rush towards the space that links the plasma membrane with vitelline envelope, inducing the envelope to undergo expansion, as well as transforming it as a fertilization envelope. Next key protein secreted by the cortical granules is the peroxidase enzyme, which in Figure 5.17 protein participation in CA2 plus entry into sea urchin eggs model for egg activation via calcium influx mammals internal fertilization. Deposition in the vagina, of time too short, is achieved by the most Olympian sperm that relies on its own flagellar power. Story 1995. Still, the sperm is transported towards the oviduct by way of the uterus muscular activity. Sperm reataxis. Sperms in turn obtain longer distance directional hands from that liquid flow from the oviduct towards the uterus. Sperms routinely exhibit reataxis, which means they migrate in the opposite direction of the flow and they use cats for calcium channels, like sea urchin sperm, to sense calcium influx and to monitor the current's direction. Median Clapham 2013. Such sperm reataxis has been observed in both mice and humans. Capacitation. Newly ejaculated sperm could not fertilize an egg. They are indeed immature. The oviduct cells in turn induce the maturation of the sperm, while mammalian sperm complete their development within the female oviducts. Chong 1951, Austin 1952. This maturation is called capacitation, the gain of capacity. The capacities of sperm gain are those for the sake of white circle recognizing the clues that guide there. Movement toward the egg white circle undergoing the acrosome reaction, white circle fusing alongside the egg cellular membrane. Sperms that have not capacitated will be held up in the cumulus matrix and are not able to reach the egg. Here, again, we see that the oviducts are not passive tracks through which sperm race. Capacitation is probably initiated by way of an EFF flux of cholesterol from out of the sperm cell membrane, which is an EFF flux generated by proteins present within the reproductive tract of female. Figure 5.18. The other steps remain the same, yet few additional steps are involved, namely translocation and capacitation. Translocation. The female reproductive tract must facilitate the meeting of sperm and egg. To position the gametes in the right place at the right time, various mechanisms are used. A mammalian oocyte has just been released from the ovary and is surrounded by a matrix of cumulus cells. If this matrix is removed experimentally or otherwise significantly altered, the fimbriae at the upper end of the oviduct will not pick up the oocyte cumulus complex, and the complex will not be able to enter the oviduct. Talbot et al. 1999. Once the oocyte cumulus complex is picked up, a combination of ciliary beating and muscle contractions transports it to the proper position in the oviduct for fertilization. The movement of sperm from the vagina to the oviduct involves several steps that occur at different times and locations. Sperm motility. Motility flagellar action is most likely necessary for sperm to pass through the cervical mucus and into the uterus. Interestingly, in those mammals where the female is promiscuous, meaning with several males in rapid succession, sperm from the same male frequently form trains, otherwise aggregates, in which the combined propulsion of the flagella makes the sperm swim faster. This strategy probably evolved for sake of competition among males. In those species without female promiscuity, the sperm usually remain as individuals. Fisher and Hoekstra 2010, Foster and Peasley 2010, Fisher et al. 2014. Uterine muscle contractions. Sperm are localized in the oviducts of hamsters, mice, cows, guinea pigs, and humans. After 30 minutes of sperm, these changes lead to unmasking of receptors present in the sperm. These unmasked receptors allow the binding with the zona pellucida while preparing the acrosome for sake of the acrosome reaction. Figure 5.18 mammalian sperm capacitation. Hyperactivation. Toward the end of capacitation, sperm become hyperactivated and they swim at a higher velocity that indeed produces greater forces. Hyperactivation is mediated by the opening of the sperm-specific calcium channels, the CASPR proteins, located within the tail of the sperm. Wren et al. 2001, Key et al. 0.2007. The symmetrical flagellum bead is altered into a rapid and asynchronous bead with a higher tendency of bending. The power of the bead in the direction of sperm head movement are thought to release the sperm from their binding with the oviduct epithelial cells. Indeed, only those hyperactivated sperms are noted to make their journey towards the egg. Suarez 2008a, b. Hyperactivation might induce the sperms to respond in a different manner to the fluid current. Uncapacitated sperm will actually make a movement in a planar direction that allows still more time for the sake of visibility for sensing temperature difference and to preferentially swim from a cooler towards a warmer site. Stern as remotaxis is noted only among the capacitated sperm. Recognition of the zona pellucida. Once within the cumulus, the sperm can make contact with the zona pellucida, the egg's extracellular matrix. The zona pellucida of mammals has a role similar to the vitline envelope amongst invertebrates. The zona is relatively far thicker and denser structure compared to the vitline envelope. The mouse zona pellucida consists of 3 major glycoproteins, namely ZP1, ZP2, and ZP3, zona proteins 1, 2, and 3 alongside accessory proteins, which in turn bind to the zona's integral structure. The human zona pellucida contains 4 major glycoproteins. They are ZP1, ZP2, ZP3, and ZP4. The sperm binding with zona is crystal clearly species specific. Also, the species might use numerous mechanisms for achieving this binding. The sperm head to attach to the epithelial cells of the oviduct. Capacitated sperm will rotate along its long axis, possibly increasing the sperm detachment from that of the epithelium. Median Clapham 2013. Once the sperm reaches the oocyte cumulus complex, hyperactivation, in conjunction with a hyaluronidase enzyme on the sperm cell membrane, allows the sperm to digest a path through cumulus cells extracellular matrix. Lin et al. 1994, Kimura et al. 2009. Thermal and chemical gradients. Heat serves as a clue. There is a thermal gradient of 2 degrees Celsius linking the distance of the oviduct and the warmer ampulla. Bahat et al. 2003, 2006. When a sperm reaches the oocyte cumulus complex, hyperactivation combined with a hyaluronidase enzyme on the sperm cell membrane allows the sperm to digest a path through the cumulus cells extracellular matrix. Lin et al. 1994, Kimura et al. 2009. Figure 5.19 steps involved in fertilization. Figure 5.20 steps involved in polyspermy prevention. Human fertilization. Key points. Human sperm has CASPR, a sperm cation channel in matured sperm. Actin polymerization and depolymerization happens in capacitation and acrosomal reaction, respectively. Molecules on sperm surface include fertilin, azumo, and cyrex are essential to facilitate the fusion with egg. Within the egg, CD9 with integrins and GPI mediate fusion. Double fertilization. Subsequent to pollen deposition upon the stigma, it is expected to germinate and grow by the style for reaching the ovule. The microspores, known as the pollen, have 2 cells, a pollen tube cell and a generative cell. The pollen tube cell will grow into a pollen tube by which the generative cells will actually travel. The pollen tube germination demands oxygen, water, and key chemical signals. When it continues to travel by way of the stylus to make a reach towards the embryo sac, the pollen tube's growth will be indeed supported by way of the tissues of the style. In the course of this process, if all the generative cell is already being split into two cells, it can now indeed divide forming two sperm cells. The pollen tube is guided by way of the chemicals which are secreted by synergids that are located in the embryo sac. It actually enters into the ovule sac by way of the microovule. Amongst the two sperm cells, one sperm will fertilize the egg cell to form a diploid zygote, while the other sperm will fuse with the two polar nuclei to form a triploid cell which in turn forms the endosperm. In angiosperms, all put together, and these two fertilization events are termed as double fertilization. Once fertilization is nearing completion, none of the other sperm can enter. The ovule which is fertilized will form the seed, whilst the ovary tissues will become the fruit, routinely enveloping the seed. Subsequent to fertilization, the embryonic development begins. The zygote actually divides, generating two cells, the upper cell known as terminal cell and the lower cell known as basal cell. The suspensor is formed by the division of the basal cell, which eventually connects to the maternal tissue. The suspensor allows nutrition to be transported from the mother plant to the developing embryo. The terminal cell divides as well, producing globular-shaped proembryo. Within dicots, popularly known as eudicots, the developing embryo will have a heart shape owing to the occurrence of the two rudimentary cotyledons. Amongst non-endospermic dicots, like Klebsiella bursa, the endosperm will develop initially, yet it will be later digested. For such cases, the food reserves will be transported towards both the two cotyledons. The embryo and cotyledons will enlarge, while they will become crowded inside the developing seed, and they will be forced to make a bend. Ultimately, the embryo and cotyledons will fill up the seed. At this point, the seed will be ready for sacred dispersal. After some time, embryonic development is halted. Growth resumes only when the seed germinates. Until the first set of leaves begins photosynthesis, the developing seedling will rely on the food reserves stored in the cotyledons. Figure 5.21 double fertilization. One sperm fertilizes egg to form two end zygote, while other sperm fuses with two polar nuclei to form three end endosperm. Figure 5.22 early development in selected organisms. Early development in sea urchin. Sea urchins show radial holoblastic cleavage. The cleavages of the first plane and the second are both meridional and are indeed perpendicular to each other. The cleavage furrow will pass by way of the animal and vegetable poles. The third cleavage acts at the equatorial plane, perpendicular to that of the earlier two cleavage planes. It also separates the animal and vegetable hemispheres from each other. The fourth cleavage is still quite distinct from the first three. The four cells located within the animal tier divide meridionally to produce 8 blastomeres, which are interestingly of equal volume. These cells are referred to as mesomers. The vegetal tier will undergo an unequal cleavage in the equatorial plane, resulting in 4 large cells known as macromers and 4 smaller micromers known as micromers located at the vegetable pole. Figure 5.23 sea urchin blastomere. The sea urchin blastula stage begins at the 128 cellular stage of development. These cells will then form a holosphere that wraps around the central cavity, known as a blastocele. Now, all of the cells will be the same size. Interestingly, the micromers have slowed cell division. Each cell remains in contact with the blastocoele's proteinaceous fluid on the inside, as well as the hyaline layer on the outside. Tight junctions connect the previously loosely connected blastomeres into a seamless epithelial sheet that completely encircles the blastocele at this point. The blastula remains one cell layer thick as the cells divide, thinning out as it expands. This is accomplished through blastomere adhesion to the hyaline layer and an influx of water that expands the blastocele. Such rapid and invariant cellular cleavages will last until the 9th or 10th cellular division, depending on the species. Following that, there is a mid-blastular transition, during which the sync of cellular division ends. New genes become active and several non-dividing cells develop cilia on their outer region. Within the fertilization envelope, the ciliated blastula begins to rotate alongside. The differences in the cells will be visible soon. The cells of the blastula's vegetable pole begin to thicken, resulting in the formation of a vegetable plate. The animal half cells will synthesize and secrete a hatching enzyme that will digest the fertilization envelope. As a hatched blastula, the embryo can now swim freely. The fate map of the sea urchin embryo was created by observing each cell layer and the type of its descendants. Furthermore, current research has further refined these maps by modifying the fates of cells injected alongside fluorescent dyes such as D. These studies show that the majority of embryonic cellular fates are not specified in the 60 cellular stage, but these cells are not irreversibly committed. The endoderm, the coelom also known as the body wall, and secondary mesenchyme, which encompasses immunocytes, pigment cells, and muscle cells. The first layer tier of micromers produces primary mesenchyme cells, which form the larval skeleton, whereas the second layer tier produces coelom cells. Even though early blastomeres have consistent fates within the larva, the majority of these fates are achieved through conditional specification. Skeletogenic micromers are cells whose fates are determined solely by them. If such micromers are isolated from the embryo and kept within the test tubes, they will generate skeletal spicules. Furthermore, if such micromers are transplanted into the blastula's animal region, their descendants will not only form skeletal spicules, but they will also change the fates of nearby cells by triggering a secondary position for the sake of gastrulation. Figure 5.24 fate map of sea urchin blastomere. The animal half of the embryo is constantly producing ectoderm, which gives rise to the larval skin and neurons. The veg 1 layer produces cells that can enter either the ectodermal or endodermal organs. The veg 2 layer will produce cells that will give rise to 3 distinct structures. Figure 5. 25 micromers ability to induce presumptive ectodermal cells to take on other fates. A. The development of a 60-cell sea urchin embryo, depicting the fates of various layers. B. A separated animal hemisphere appears as a ciliated ball of indifferentiated ectodermal cells, which is known as a Dower-glyla. C. When an isolated animal hemisphere is combined with isolated micromers, a well-recognizable pluvius larva is formed. Figure 5. 26 ability of micromers to induce a secondary axis in sea urchin embryo. A. Micromers are being transplanted from a 16-cell embryo within the vegetable pole of an animal pole of a 16-cell embryo host. B. The micromers that were transplanted invaginates into the blastocele for creating a newer set of skeletogenic mesenchyme cells, which induces the animal pole cells located adjacent to them to form the vegetable plate endoderm cells. C. The micromers that are transplanted forms the skeletal rods, whereas the induced animal cap cells forms the secondary archenteron. Cells which routinely generate ectodermal skin cells will be redefined as endoderm, which in turn would generate a secondary gut. The micromers appear to generate a signal that tells the cells adjacent to them to become endoderm and induces them to invaginate into the embryo. Their potential towards reorganizing the embryonic cells is well pronounced if all the isolated micromers were recombined with an animal cap isolated. The top two animal tiers, the animal cap cells will originally generate endoderm and more or less normal larva will be developing. Within a normal embryo, the veg 2 cells will become specified by way of the micromers and they in turn will help to specify the veg 1 layer. In the absence of the veg 2 layer, the veg 1 cells will be able to generate endoderm, yet the endoderm is not specified as mid-gut, foregut, or hindgut. Thus, there appears to be a cascade wherein the vegetable pole micromers induce the cells above them to become the veg 2 cells and the veg 2 cells induce the cells above them to assume the veg 1 fates. Thereby, the micromers will undergo the process of autonomous specification for becoming the skeletogenic mesenchyme, and such micromers will generate the initial signals that specify the other tiers of cells. The identities of these signaling molecules that are involved in these steps are now recognized and identified. The molecule which is much accountable for sake of specification of the micromers also, their ability towards inducing the neighboring cells looks to be beta-catenin. Established within the egg cytoplasm ahead of fertilization. The axis of animal vegetable pole structure is the futuristic anterior-posterior axis with the vegetal region, which sequesters the maternal parts that are important for sake of posterior development. In the majority of sea urchins, the axis of dorsal ventral and left right will be specified only after fertilization, yet their specification behavior is not yet well understood. Since the plane of first cleavage could be perpendicular, parallel, or oblique alongside the dorsal ventral axis, it is possible that the dorsal ventral axis is not yet being specified till the 8th cellular stage when the cell boundaries mark these axis positions. Sea urchin gastrulation. The late sea urchin blastula is made up of a single layer of approximately 1,000 cells that form a hollow ball that is flattened at the vegetal end. The blastomeres, which are derived from different regions of the zygote, will vary in size and properties. The fate of the blastula as it develops varies according to region. The development of a sea urchin begins with gastrulation and continues to the pluvius larva stage. Only through the movements that occur during gastrulation can the cellular fate of each cell layer be determined. The beta-catenin serves as a transcription factor, which is frequently activated by way of the WNT pathway, as various evidences indicate that it is for the sake of this role. Foremost, in due course of normal development of sea urchin, beta-catenin begins its accumulation inside the cell nuclei, which is destined for becoming endoderm and mesoderm. If sea urchin embryos are treated with lithium chloride, it triggers the beta-catenin accumulation within their cells. Also, such treatments will transform the presumptive ectoderm to endoderm. By contrast, the experimental processes which inhibit the accumulation of beta-catenin within the vegetal cell nuclei will block the endoderm and mesoderm formation. It is indeed probable that the levels of accumulation of nuclear beta-catenin will lead in determining the mesodermal and endodermal fates of the vegetal cells. Next, beta-catenin is very profound for the sake of providing the micromers with their inductive ability. Micromers derived from the embryos where beta-catenin was being inhibited from reaching the nucleus will be unable to induce the animal hemisphere cells towards forming an endoderm, whilst the second axis will not be formed. Specification of axis. In the sea urchin blastula, the cell face will line up alongside the animal vegetal axis, which was Figure 5.27 gastrulation movement of sea urchin which leads to formation of pluvius larva. Growth. The tip of the archendron is actively pulled to the blastoceal roof by the phylopodia of the non-skeletogenic mesenchyme cells during the final stage of invagination. Early development in tunicates have bilateral holoblastic cleavage. The skeletogenic mesenchyme ingresses through an epithelial-mesenchymal transition in which these cells lose coherence while gaining affinity to adhere to the matrix within the blastoceal. Cell shape changes, cell proliferation, and convergent extension all work together to coordinate archendron invagination and Figure 5.28 bilateral symmetry in the egg of the tunicate. A. Uncleaved egg. The regions of cytoplasm destined to form particular organs. B. 8 cell embryos showing the blastomeres and fates of various cells. C. Deep views of later embryos from the vegetable pole. Indeed, every cell division is asymmetrical from the 8 to 64 cell stages, with posterior blastomeres always being smaller than anterior blastomeres. Nishida 2005, Started et al. 2007. The posterior centrosome in the blastomere migrates toward the centrosome attracting body cap, a macroscopic subcellular structure composed of endoplasmic reticulum, prior to each of these unequal cleavages. Figure 5.29 cytoplasmic rearrangement in the fertilized egg. A. Prior to fertilization, the gray yolk constituting the inner cytoplasm is wrapped by yellow cortical cytoplasm. B. Following sperm entry in the middle of the oocyte's vegetal hemisphere, the yellow cortical cytoplasm and clear cytoplasm derived from the breakdown of the oocyte nucleus contract vegetally towards the sperm. C. As the sperm from nucleus migrates anally towards the newly formed. D. The last position of yellow cytoplasm indicates the location of cells that give rise to tail muscles. And TBX6 appear to activate most likely in a feed-forward manner the muscle-specific gene snail. Autonomous specification of endoderm, beta-catenin. Presumptive endoderm originates from the vegetal A4.1 and B4.1 blastomeres. The specification of these cells coincides with the localization of beta-catenin. Recall that beta-catenin is also involved in endoderm specification in the sea urchin embryo. Endoderm in the tunicate embryo is lost when catenin is inhibited, and it is replaced by ectoderm. Amai et al. 2000. Increasing catenin synthesis, on the other hand, causes an increase in the endoderm at the expense of the ectoderm, just as in sea urchins. The catenin transcription factor appears to work by inducing the synthesis of the homeobox transcription factor LHX3. Endoderm differentiation is prevented when the LHX3 message is inhibited. Cito et al. 2001. Conditional specification of mesenchyme and notochord by the endoderm. Although the majority of tunicate muscles form autonomously from the yellow crescent cytoplasm, the most posterior muscle cells form through conditional specification through interactions with the descendants of the A4.1 and B4.2 blastomeres. Nishida 1987, 1992a, b. Furthermore, inductive interactions form the notochord, brain, heart, and mesenchyme. Indeed, FGF secreted by endoderm cells appear to induce the notochord and mesenchyme. Nakatani et al. 1996, Kim et al. 2000, Amai et al. 2002. These FGF proteins stimulate the expression of brachyury, a protein that binds to the cis-regulatory elements that regulate notochord development. Davidson and Christian 2006. The breakdown of the nucleus during meiosis produces a clear substance that accumulates in the animal hemisphere of the egg. Within 5 minutes of sperm entry, the interclear and cortical yellow cytoplasms of the egg contract into the vegetal lower hemisphere. Proton et al. 2005, 2008, Started et al. 2005. The yellow lipid inclusions migrate alongside the male pronucleus as it migrates from the vegetal pole to the equator of the cell along the future posterior side of the embryo. This migration forms the yellow crescent, which extends from the vegetal pole to the equator and generates the majority of the tunicate larva's tail muscles. These cytoplasmic regions move due to the organization of microtubules by the sperm centriole and a wave of calcium ions that contracts the animal pole cytoplasm. Sawada and Chatham 1989, Speck Snyder et al. 1990, Robiers et al. 1995. Autonomous specification of the myoplasm, the yellow crescent in macho -1. When yellow crescent cytoplasm is experimentally transferred from the B4.1 muscle-forming blastomere to the B4.2 otherwise a 4.2 ectoderm-forming blastomeres of an 8-cell tunicate embryo, the ectoderm-forming blastomeres generate muscle cells in their normal ectodermal progeny. Nishida and Sawada 2001 discovered that this muscle-forming determinant was an mRNA that encoded the transcription factor macho-1. The macho-1 protein is a transcription factor that is required for the activation of several mesodermal genes, including TBX6, snail, and muscle actin and myosin genes. Yagi et al. 2004, Sawada et al. 2005. Only the TBX6 protein phenotype mimicking macho-1 function by inducing ectopic muscle differentiation. Macho-1 appears to directly activate a set of TBX6 genes, and TBX6 proteins appear to activate the rest of muscle development. Yagi et al. 2005, Kugler et al. 2010. Both macho-1 figure 5.30 the two-step steps for sake of specifying the marginal cells of tunicate embryo. The first step involves the cell's acquisition of the macho-1 transcription factor. The second step involves the reception of the FGF signal from the endoderm. Figure 5.31 gene network leading to notochord development in early tunicate A beta-catenin accumulation leads to expression of FOX gene. FOX protein helps cells to become endoderm to secrete FGFs. B FGFs induce brachyury expression in neighboring cells. These are the cells that will become notochord. Early development of zebrafish. Zebrafish cleavages, yolking out the process. The eggs of most bony fish are telelecithal, meaning that most of the cytoplasm is occupied by yolk. Cleavage could occur only within the blastodysk, which serves as a thin region of cytoplasm, which is yolk-free, located at the animal pole region. The cellular divisions indeed do not totally split the egg. Thereby, this cleavage type is known as meroblastic, Greek meros, heart. Only the blastodysk will become the embryo, while such type of meroblastic cleavage is usually termed as discoidal. The initial cell divisions will adopt a highly reproducible pattern like that of meridional and equatorial cleavages. Such divisions will be rapid, which takes roughly about 20 minutes for each division. The first 10 divisions will occur synchronously, leading to formation of a cell mound that indeed sits at the animal pole region within a large yolk cell. Such cell mounds make up to form the blastoderm. Initially, all of the cells will maintain a certain open connection alongside one another and along the yolk cells that underlie, such that averagely sized 17 kDa molecules could pass indeed freely from one blastomere to that of the next. King and Kimmel 1993. Noticeably, as and when the daughter cells migrate a bit away from each other, they still will retain such bridges by way of long tunnels that connect the cells. Canacaro et al. 2011. Fish embryos, like many other embryos, undergo a mid-blastular transition seen around the 10th cell division in zebrafish. When zygotic gene transcription begins, cell division slow, and cell movements become evident. King and Kimmel 1993. Maternal to zygotic transition requires the function of maternally deposited microRNAs near 430, for sake of instance, which progressively degrade maternal messages and thereby foster a fast transfer of transcriptional responsibilities to the zygotic genome. At this same time, 3 distinct cell populations could be distinguished. The priority of these is the yolk syncatial layer, also known as YSL, Agassiz and Whitman 1884, Carvalho and Heisenberg 2010. The yolk syncatial layer will not contribute cells otherwise nuclei of the embryo, yet it is critical for sake of otherwise producing the organizer of the fish, which in turn patternizes the mesoderm and leads the ectodermal epithelial over the embryo. The YSL is being formed at the 10th cell cycle, where the cells located at the blastoderm vegetal edge will fuse with the yolk cell that underlies. Early development in mammals. Mammalian cleavage. Prior to fertilization, the oocyte of mammalian cells will be wrapped up within the cumulus cells and will be released from out of the ovary while it is swept away by the fimbriate to the oviduct. Fertilization occurs in the amphipolar region of the oviduct, which is located close to the ovary. After sperm entry, meiosis is completed and the first cleavage occurs about a day later. The position of the first cleavage plane may be affected by the point of sperm entry. Pietrowska and Zernicka gets 2001, and in mice, a sperm-borne microRNA, mRNA 34C, is required to initiate this first cell division. This mRNA appears to both bind and inhibit BCL2, a protein that prevents cells from entering the S phase of the cell cycle. Liu et al. 2012. The two nuclei produced by this cleavage include the first nuclei, which contain the entirety of its genome content. When haploid pronuclei meet mammalian embryos, they gate cellular division. The first cleavage is a normal meridional division. However, in the second cleavage, one of the two blastomeres divides meridionally while the other divides equatorially. This is known as rotational cleavage. Another significant difference between mammalian and anemio cleavage is the marked asynchrony of early cell division. Mammalian blastomeres do not divide all at once. Thus, despite the fact that mammalian embryos do not grow exponentially from 2 to 4 to 8 cells, they frequently contain odd numbers of cells. Furthermore, unlike the genomes of rapidly developing animals, the mammalian genome is activated during early cleavage, as zygotically transcribed proteins are required for cleavage and development. Compaction. Compaction is one of the most important events in mammalian cleavage. As the mouse blastomeres reach the 8 cellular stage, they adapt to a spaced arrangement. Following the third cleavage, the blastomeres will reform into a spectacular behavior change. Cell adhesion proteins like that of E-cadherin will be well expressed as the blastomeres gradually huddle together and form a cellular ball of compact nature. Carrieres et al. 1983. Fleming et al. 2001. This tightly packed arrangement is stabilized by tight junctions that create the linking amongst the outside cells, that in turn seals off the inside region of the cellular sphere. The cells within the sphere form gap junctions, allowing small molecules and ions to pass through and connect them, much like in the fish's early blastula. The cells of the 8-cell compacted embryo divide to form a 16-cell morula. The morula encompasses a tiny group of internal cells which are surrounded by big-sized external cells. Barlow et al. 1972. Majority of the external cells' descendants will in turn form trophoblasts protector, whilst the internal cells will in turn give rise to the intercellular mass shortly known as ICM. The intercellular mass in turn will form the embryo, becoming localized upon one side of the trophoblast ring cells, while the resulting blastocyst serves as a hallmark of mammalian cleavage. The trophoblast cells do not form embryonic structures, but rather the tissues of the chorion, the extraembryonic membrane, and the portion of the placenta that allows the fetus to get oxygen and nourishment from the mother. The chorion also secretes hormones that cause the mother's uterus to retain the fetus and immune response regulators that ensure the mother does not reject the embryo. It is critical to remember that one of the most important outcomes of these first divisions is the formation of cells that attach the embryo to the uterus. As a result, the formation of the trifectoderm is the primary differentiation event occurring during mammalian development. The earliest blastomeres, for example, each blastomere in a 2-cell embryo can form both trophoblast and neuroblast. Intercellular mass precursor cells and embryo precursor cells. These cells are referred to as totipotent, which means capable of everything in Latin. The cells of the intercellular mass are referred to as pluripotent, Latin for capable of many things. That is, while each ICM cell can generate any cell type in the body, it can no longer form trophoblasts. These pluripotent cells of the inner cell mass can be isolated and cultured, where they will self-renew indefinitely, maintain their pluripotency and thus acquire the characteristics of embryonic stem cells under the right conditions. Figure 5.32 transcriptional core circuitry for the ICM cells pluripotency nature. A circuit where October 4 SOX2 dimers will activate the NANOG genes. NANOG protein in turn activates its own gene and genes promoting pluripotency. B, the interconnected regulatory circuit whereby October 4 SOX2 and NANOG each activate their own and one another synthesize. Cavitation. The embryo proper within mice is being obtained from the intercellular mass located within the 16-cellular stage, which is being supported by way of cells that divide from the cells localized outside the morula in due course of the transition towards the 32-cellular stage. Peterson et al. 1986, Fleming 1987, McDowell et al. 2011. The intercellular mass gives rise to the embryo and its associated yolk sac, allantois, and amnion. The ICM comprising approximately 13 cells at that stage, and the trophoblast cells have separated by the 64-cell stage and neither contribute cells to the other group. Dice et al. 1987, Fleming 1987. In turn, the ICM actively supports the trophoblast by secreting proteins that stimulate trophoblast cell division. Tanaka et al. 1998. The morula, on the other hand, lacks an internal cavity. However, during a process known as cavitation, the trophoblast cells secrete fluid into the morula, resulting in the formation of a blastocele. Sodium pumps NOT plus K plus ATPase, and the NOT plus H plus exchanger in trophoblast cell membranes pump NOT plus into the central cavity. The subsequent accumulation of NOT plus attracts water osmotically, both creating and expanding the blastocele. Borland 1977, Eckert et al. 2004, Kawaguchi et al. 2004. The oviduct cells, which transport the embryo to the uterus, initiate the sodium pumping activity. Chu et al. 2004. The inner cell mass of the blastocele becomes positioned on one side of the ring of trophoblast cells as the blastocele expands, resulting in the distinct mammalian blastocyst. Mammalian gastrulation. Mammalian gastrulation is not unlike that of birds. There are two signaling centers and one is located in the node while the other one is in the visceral endoderm anteriorly. The latter center is critical for sake of establishing the body axis, while the former is critical in inducing the nervous system and in patterning axial structures caudally from the midbrain. Hox genes pattern the anterior posterior axis, as well as help specify positions alongside that axis. If Hox genes are knocked out, segment-specific malformations could arise. Likewise, triggering the Hox gene's ectopic expression can in turn alter the body axis. The homology of gene structure links the expression patterns that link Drosophila with mammalian Hox genes, which indicates that such a mechanism of patterning is ancient. The mammalian left-right axis is specified similarly to that of the chick, yet with some significant differences in the roles of certain genes. In amniogastrulation, the pluripotent epithelium and epiblast generates the mesoderm and endoderm, which migrate through the primitive streak, as well as the precursors of the ectoderm, which remain on the surface. By the end of gastrulation, the head and anterior trunk structures are formed. The elongation of the embryo prevails by precursor cells within the caudal epiblast, which in turn surrounds the posteriorized Hensen's node. In each class of vertebrates, a neural ectoderm is allowed to form, whereas the BMP-based epidermal tissue induction is prevented. Fraternal twins arise from two separate fertilization events. Identical twins in turn formed by embryo splitting. It forms two cellular groups in due course of stages while there are still pluripotent cells within the embryo. Experimental evidence suggests that conjoined twins might occur through the formation of two organizers within a common blastodisc. Cleavage and gastrulation. Cleavage and types. The steps of cleavage serve as one of the three mechanical activity within the transformation of a unicelled egg into a multicellular embryo. It is triggered by the sperm in due course of fertilization. However, amongst parthenogenetic eggs, the cleavage will start alongside, which was not influenced by fertilization. The cleavage steps of cellulation take place by way of continued meiotic divisions. These divisions in turn result in formation of cells called blastomeres. In developmental end stages, the blastomeres will occupy various regions and differentiate into several types of body cells. The frog's egg undergoing the first cleavage was noticed by Swammerdam in 1738. The whole process of cleavage in a frog's egg was studied by Pregos with Dumont in 1824. By way of the microscope, development of cleavages with still more stages were in turn observed amongst the sea urchins' eggs, amphioxus, starfishes, along with hens' eggs. Within these studies, it was evident that all the divisions within cleavage were meiotic. The meiotic steps were in turn faster. Within the sea urchin, eggs of cellular division of the blastomeres were noted every 30 minutes. The size of the resultant daughter cells, known as blastomeres, decreases as the cleavage progresses. The blastomeres do not grow during cleavage. The embryo's total size and volume remain unchanged. The cleavages produce a compact mass of blastomeres known as morula. It transforms into blastula. While the blastula's wall is known as the blastoderm, the central cavity is known as the blastocele. Planes of cleavage. An egg could be divided from different planes during cleavage. Based on the position of the cleavage furrow, the planes of cleavage are named. Meridional plane. The plane of cleavage lies on the animal vegetal axis. It is both of the egg poles. Thereby, the egg is now being compartmented to form two equal halves. Vertical plane. The cleavage partition may be present on meridional planes either sides. The furrows indeed will move from the animal towards the vegetal pole. The divided cells may be non-equivalent in size aspect. Equatorial plane. Such cleavage plane will bifurcate the egg exactly at right angles from that of the critical main axis. It relies upon the equatorial plane. It creates two equal halves of the egg. Latitudinal plane. It is indeed like that of the equatorial plane, yet it relies upon the both sides of the equator. It is known popularly as transverse, otherwise horizontal cleavage. Yolks roll on cleavage. Yolk is indeed required for the development of embryo. But still, the egg that was previously fertilized must follow sequential steps for undergoing stepwise developmental stages that in turn will result in a young form, which will initiate the subsequent generation. Anyhow, with all the role of yolk in the developmental procedures are so adapted and modified that a well-formed embryo is obtained. The initial role taken by yolk is noticed during the cleavage process. The volume of the yolk and its distribution will in turn influence the process. As a result, various cleavage patterns are being recognized. Cleavage of holoblastic nature. With such nature of the cleavage, the furrow will divide the whole egg. This cleavage will be equivalent else non-equivalent. White circle equal nature of holoblastic type of cleavage. Amongst the microlecythal type and isolecythal type of eggs, such cleavage will mark way towards blastomeres production with equal size. For instance, amphioxus in placental mammals. White circle unequivalent nature of holoblastic type of cleavage, noted in mesolecythal and telolecythal eggs, where the cleavage will lead towards the production of unequivalent sized blastomeres. Blastomeres of very smaller size were termed as micromers, while the larger ones and a few of large size are popularly known as macromers. Next type is of meroblastic cleavage. Within this type, the cleavage furrow formation will be restricted towards the active region of the cytoplasm localized within the animal pole, which is commonly termed as macrolecythal egg. The yolk superficially surrounds the egg, leading to formation of centrolecythal eggs. Meroblastic cleavage might be of two types. White circle discoidal cleavage. Due to the fact that the macrolecythal eggs will have an enormous quantity of yolk, the cytoplasm is now being restricted towards a small region within the animal pole. Thereby, the cleavage furrow is produced only within the animal pole like a disc. Thereby, these cleavage patterns are commonly known as discoidal meroblastic cleavage. For instance, it's observed among birds and reptiles. White circle superficial type of cleavage. Amongst central eggs, the pattern of cleavage is restricted towards the cytoplasm of periphery within the egg. Noted amongst insects. Laws of cleavage. There appear to be several cleavage patterns. However, all cleavages follow the same pattern. Certain basic principles laws govern the cleavages. Sachs laws. Sachs proposed these laws in 1877. Cells usually divide into identical daughter cells. Every newer division plane will have a 90 degrees intersection with the next plane. Balfour's law, Balfour 1885, states that the speed or rate of cleavage in any region of the egg is inversely proportional to the amount of yolk it contains. Different types of blastula. The blastula of various groups of animals differs in form and structure depending upon a variety of factors, such as the size of the amount and distribution of yolk, etc. The following blastula categories have been identified in various animal groups. Coeloblastula is a hollow blastula with a large and spacious blastocele. Typically, the blastocele is filled with mucopolysaccharide containing fluid. The blastula formed by holoblastic equivalent cleavage, as seen in echinoderms and amphioxus, is known as equivalent coeloblastula. The blastoderm in this case is single-layered. As in frogs, holoblastic unequivalent cleavage results in unequivalent coeloblastula. It has a blastocele that is displaced toward the animal pole in a multi-layered blastoderm. Gastrulation. It is a complex series of cell movements that rearranges cells, providing them with new neighbors and beat these changes place cells in a new environment and give them the ability to receive new signals. Seed results in the formation of the 3 germ layers that will make up the majority of the subsequent embryo, ectoderm, endoderm, and mesoderm. The following types of morphogenic movements have been identified. Individual cells pass by. Migration is the movement of individual cells over other cells in a matrix. Ingression is the movement of individual cells or small groups of cells from an epithelium into a cavity. Cells move in groups. Invagination is defined as the local inward buckling of an epithelium. Involution is the inward movement of a cell layer around the point or edge. Apoboly spread of an outside cell layer to envelop a yolk mass that would otherwise be in a deeper layer. Delamination is the process of dividing a single cell sheet into 2 otherwise parallel sheets. Convergent extension is the lengthening of a cell layer in one dimension while shortening it in another. Figure 5.33 gastrulation types and examples. In due course of development, there will be 3 layers of germ cells. Ectoderm encompasses the formation of nervous tissue alongside skin epidermis with sensory organs. Endoderm encompasses the lining of gut tract along with digestive glands, also seen amongst lining of respiratory tract. Mesoderm encompasses the connective tissues, which make up the circulatory system alongside tendons, bones, and ligaments. Dermis encompasses the skin alongside the excretory structures, reproductive structures, and muscle. Early Drosophila development. The sperm enters an egg that is already activated. Activation of eggs within the Drosophila is being accomplished at the stage of ovulation, which routinely occurs a few moments ahead of commencement of fertilization. Whenever the oocyte of Drosophila is being squeezed up by way of narrow orifice, there will be opening up of calcium channels, which makes the inflow of Ca2+. The nucleus of oocyte will regain the meiotic divisions where the cytoplasmic mRNAs will be translated without the events of fertilization. There is only one site where the sperm could enter the egg, forming the micropyle, which serves as a tunnel within the chorion eggshell located at the future dorsal anterior region of the embryo. The micropyle allows sperm to pass through it one at a time, preventing polysporny in Drosophila. Furthermore, despite cortical changes, cortical granules are not available for blocking up polysporny. The moment when sperm enters the egg, the specification of the body axis will commence, thereby implying that the egg is organized priorly as an embryo. The sperm will enter through the intact egg instead of fusion between egg and sperm cellular membranes. Before the pronucleate fuse, the DNA of the male and female pronucleate replicates, and after the pronucleate fuse, the maternal and paternal chromosomes remain separate until the end of the first mitosis. Cleavage. Majority of insect eggs adapt to superficial cleavage, where a huge yolk will be placed centrally, making up the confinement of cleavage towards the egg cytoplasmic edge. One of the fascinating features of this cleavage pattern is that cells do not form until after the nuclei have undergone multiple rounds of cellular divisions. In the Drosophila egg, karyokinesis nuclear division occurs without cytokinesis cell division, so as to create a syncytium, which remarks a singular cell with several nuclei that remain localized within a unified cytoplasm. The zygote nucleus will have multiple rounds of karyokinesis within the central region of the egg. Almost 256 nuclei will be produced by way of 8 rounds of nuclear divisions with a time frame of 8 minutes for every division. This rapid rate of division is accomplished by repeated rounds of alternating S-DNA synthesis and M phases, remarking the meiotic division. This phase totally skips G phases, remarking gap phase of the cell cycle. Approximately 5 nuclei reach the surface of the embryo's posterior pole during the 9th division cycle. These nuclei are surrounded by cell membrane and produce the pole cells that give rise to adult gametes. At cycle 10, the other nuclei migrate to the cortex periphery of the egg, and mitosis continue, albeit at a slower rate. Foe et al. 2000. During such nuclear division stages, the embryo is referred to as a syncatial blastoderm because none of the cellular membranes will survive apart from the egg itself. When the nuclei reach the egg's periphery during the 10th cleavage cycle, they are surrounded by microtubules and actin filaments. The nuclei and their associated cytoplasmic islands called energids. Following the 13th cycle cellular division, the cellular membrane that has been wrapping the egg thus far will make an inside fold that connects the nuclei. Every energid will eventually divide, yielding single cells. These steps result in the formation of the cellular blastoderm. All of the cells will be arranged as a single layer of jacket wrapping the yolky core of the egg. Midblastular transition. The moment when the nuclei attain the peripheral region, the time frame needed for completing every round of the upcoming 4 divisions is marked by a bit of longer duration. The cycles 1 to 10 have a routine of 8 minutes for every cycle, while the 13th cycle, the final cycle within the syncatial blastoderm, will have a time of about 25 minutes for completion. The cycle of 14 makes Drosophila embryo to form cells in asynchronous form. Certain cellular groups will complete such a cycle within 75 minutes of time, while few groups will in turn have 175 minutes for completion. Fo 1989. During such time, the genes of the nuclei become active. Before this point, the early development of Drosophila is being directed by way of proteins alongside mRNAs, which are localized inside the egg in due course of oogenesis. These serve as products of the mother's genes, but do not represent the genetic makeup of the nuclei content of the embryo. The genes which remain active within the mother will induce product formation for sake of the early development of the offspring is profoundly known as maternal effect genes, whereas the mRNAs within the oocyte are termed as maternal messages. Zygotic gene transcription, i.e., the activation of the embryo's own genes, begins around cycle 11 and is greatly enhanced at cycle 14. This slowdown of nuclear division, cellularization, and key rise in new RNA transcription are routinely termed as the midblastular transition. Yuan et al. 2016. It is at this stage that the maternal mRNAs will undergo degradation, while developmental control is transferred over to be carried out by the zygote's genome. These steps of maternal to zygotic transition are noticed within the vertebrate and invertebrate phylum embryos. Figure 5.34 axis formation in Drosophila. Figure 5.35 model of Drosophila anterior posterior pattern formation. A. The pattern is established by maternal effect genes. B. Maternal effect genes. C. Gap gene protein expression as well as overlap. The first such zygotic genes to be expressed are termed gap genes as mutations in the pos gaps in the segmentation pattern. Such genes are being expressed within certain broad, 3 segments wide with partly overlapping domains. Gap genes usually encode transcription factors, where unique combinations of concentrations of gap gene proteins will in turn regulate the transcription of pair rule genes, which will divide the embryo into periodic units. The transcription of various pair rule genes will result in a pattern containing strips of 7 transfer strands that are 90 degrees to the anterior posterior axis. The transcription factors that are being encoded by way of the pair rule genes will in turn activate the segment polarity genes, for which the mRNA and protein products will bifurcate the embryo to form 14 segment wide units, which establish the embryo periodicity. While at similar time, gap, pair rule, and segment polarity genes protein products will interact for regulating one more category of genes, known as the homeotic selector genes. This gene transcription will determine the developmental fate of every segment. Table 5.5 Maternal effect genes that establish the anterior posterior polarity of the Drosophila embryo source. K.D. Anderson. 1989. In Genes and Embryos Frontiers in Molecular Biology Series. D.M. Glover and B.T. Haynes eds. pp. 1-37. IRL. New York. Figure 5.36 Syncatial specification in Drosophila. Anterior posterior specification originates from morphogen gradients in the egg cytoplasm. Figure 5.37 bicoid gene encodes the morphogen is responsible for sake of head structures in Drosophila. Figure 5.38 model of anterior posterior axis generation by virtue of maternal effect genes A. Bicoid, nonos, hunchback, and cotyledon RNAs are deposited in oocyte. Beat gradient of bicoid protein will extend from the anterior region and move towards posterior region, whereas the protein gradient of nonos will extend from the posterior region towards the anterior region. C. Parallel interactions will lead to translational gene regulation, which in turn will accomplish the anterior-posterior axis formation of the Drosophila embryo. Bicoid protein forms gradient of anterior-posterior. Hunchback protein forms gradient of anterior-posterior. Nonose protein forms gradient of posterior-anterior. Caudal protein forms gradient of posterior-anterior. Anterior-posterior axis of Drosophila's embryo is specified by means of three gene sets: organizing center anterior region, genes defining the organizing center posteriorly, and genes defining the terminal boundary regions. Organizing center anterior region. The anterior organizing center is being localized at the anterior end of the embryo and will function by way of a bicoid protein gradient. Bicoid will function as a transcription factor for activating the gap genes that are anterior specific with a translational repressor for suppressing the gap genes that are posteriorly specific. Genes defining the organizing center posteriorly. The organizing center of the posterior is localized within the posterior pole. Such a center will act by translational of nonose protein towards the inhibition of anterior region formation, while transcriptional of the caudal proteins will in turn trigger the genes forming the abdomen. Genes defining the terminal boundary regions. The boundaries of the acronym are defined by the product of the torso gene, which is activated within the embryo step. Table 5.6 Majority of genes altering the Drosophila segmentation pattern. The segmentation genes help to make the early embryo to divide and form sequence segmental primordia through the anterior-posterior axis. This really aids in the transition from specification to determination. Earlier, segmentation genes functioning were dictated by virtue of the zygotic mutations that in turn disrupts the body plan. These genes were grouped to three subcategories based upon the mutant phenotypes they express. Nusslein-Volhard as well as Weishaupt 1980. Gap mutants. This group will have contiguous segments without large regions of the body, several contiguous segments. Pair rule mutants lack the alternative segment. Segment polarity mutants exhibit defects for instance deletions, duplications, polarity reversals within every segment. Figure 5.39 segmentation gene mutations have three major types. The left side shows early cleavage alongside with region where particular gene is normally transcribed in wild-type embryo. These areas are deleted as the mutants develop into the late-stage embryos. Gap genes. In the anterior-posterior axis, the gap genes are either activated else repressed by virtue of maternal effector genes. Such expression patterns are well exposed within the embryonic regions that miss out the gap mutations. Good illustration at 4-6 pair segments by Kruppel. It serves as a key expressive gene localized within the central region of the embryos. Mutations leading to lack of Kruppel proteins will make the embryo devoid of pair segments within these locations. In the Drosophila embryo, deletion of gap gene mutants of Hunchback, Kruppel, as well as Knerps usually remarks its effects on the entire segmented region. The giant, an important gap gene, will overlap those three, as the gap gene's tails as well as Huckleberry are expressed in domains near the anterior as well as posterior ends of the embryo. Taken together, the four gap genes of the trunk have enough specificity to define a cell's location with an error of only around 1% alongside the embryo's anterior-posterior axis. Due to the unique interaction of the gap gene products, every cell gets its unique spatial identity. Homeotic selector genes. Subsequent to setting up boundaries of segmentation, gap genes as well as pair rule genes will interact for regulating homeotic selector genes, which in turn specifies key structures of every segment. Towards the completion of the cellular blastoderm stage, every region of segment primordium will get an identity by virtue of its novel combo expression of homeotic, gap, as well as pair rule gene products Levine as well as Harding 1989. Majority of the homeotic genes are present within the two regions of chromosome 3. The foremost region, popularly known as the antennapedia complex, will have the homeotic genes namely the forms DFD, antennapedia, ANTP, sex chromes reduced SCR, proboscipedia, PB, as well as labial lab. The labial as well as deformed genes specify the head segments, while sex chromes reduced as well as antennapedia makes up the identity of the thoracic segments. The proboscipedia genes are much functional in adults while if it's lacked, it will lead to transformation of mouth to legs. Figure 5.40 Drosophila, homeotic gene expression in A, homeotic genes expression in B, institute hybridization for sake of 4 genes at a later stage. Figure 5.41a, the wild-type fruit fly's wings emerges from the second thoracic segment. B, A4 winged fruit fly created by combining 3 mutations in cis regulators of the UBX gene. These mutations convert the third thoracic segment into a second thoracic segment. The bithorax complex is the second region of the homeotic genes, Lewis 1978, Maeda as well as Clark 2009. Within this complex, 3 protein coding genes are found. The ultravithorax UBX gene is required for the identity of the third thoracic segment, as are the abdominal A ABDA and abdominal B ABDB genes, which are responsible for the segmental identities of the abdominal segments. Sanchez Herrero et al. 1985. The homeotic complex, also known as home C, is a chromosome region that contains both the antennapedia complex and the bithorax complex. When the ultravithorax gene is deleted, the third thoracic segment, represented by halteres, is transformed into a second thoracic segment. As a result, the Drosophila will have 4 wings, an embarrassing situation for a classic dipteran 2-winged organism. Similarly, antennapedia protein specifies the second thoracic segment. However, whenever these flies have the antennapedia gene mutation, which causes the legs to grow from the head instead of the antenna. Figure 5.42a, head of a wild-type fruit fly B, when there is a mutation in the antennapedia gene, it leads to growth of legs in position of antenna. Axis formation. The polarity of the dorsal ventral region is triggered whenever the nucleus makes a movement towards oocyte dorsal anterior sequestering the Gerkin protein towards synthesizing proteins located within the dorsal side of the egg. The gradient expression of dorsal protein is routinely triggered within the nuclei. Such nuclei localized within the most ventral surface will transform to form mesoderm, while those at the lateral will form the neurogenic ectoderm. Organs are being formed at the intersection of dorsal ventral as well as anterior posterior regions of gene expression. Early development in frog. Majority of the frogs will adapt to external fertilization, whereby females lay the egg and males will be externally fertilizing them. Ahead of fertilization, the polarity of the egg will be fixed, making dense yolk at the vegetal bottom end and the upper half marks the animal part of the egg. It must be kept in mind that the animal part has very little yolk. Fertilization occurs most frequently in the animal hemisphere of the amphibian egg. This proclivity for sperm egg binding to occur in the animal hemisphere may be due to the spatially restricted interaction of surface lipoproteins on the sperm with the vitally envelope and cell membrane in the animal hemisphere of the egg. Nagai et al. 2009, Kubo et al. 2010. The point of sperm entry is significant owing to the fact that it marks up the polarity of the dorsal ventral region. The point of sperm entry represents the ventral, i.e., the belly region, whilst the site which is 180 degrees opposite to the region of sperm entry signifies the dorsal, i.e., the spinal side. The sperm centriole will enter the egg alongside with the sperm nucleus, taking part in organizing the egg microtubules in parallel tracks within the vegetal cytoplasm, thereby separating the outer cortical cytoplasm from the yolky internal cytoplasm. Such microtubular tracks will in turn permit the cortical cytoplasm to undergo rotation alongside with inner cytoplasm. In fact, these arrays are first spotted ahead of commencement of rotation, making up to progressively line up in due course of rotation, which disappears the moment the rotation ceases. Within the zygote, the cortical cytoplasm rotates about 30 degrees alongside with respect to the internal cytoplasm. In some cases, this exposes a region of gray-colored inner cytoplasm directly opposite the sperm entry point through 1887, in cells as well as Vinterberg 1948. This region, the gray crescent, is where gastrulation will begin, Maines as well as Linson 1980, Vincent et al. 1986. Gastrulation begins on the side of the egg opposite the point of sperm entry, and this region will become the embryo's dorsal portion. The microtubular arrays organized by the sperm centriole during fertilization help to specify dorsal to ventral cell fate and thus influence these movements. Thereby, the dorsal ventral axis of the larva can be traced back to the point of sperm entry. Said another way, the frog zygote already knows its head from its tail. Figure 5.43 reorganization of cytoplasm in the newly fertilized frog egg. Schematic cross-section of an egg midway through the first cleavage cycle. The egg contains radial symmetry about its animal vegetal axis. The sperm will enter at one side, as well as the sperm nucleus is migrating inward. The cortex represented is like that of Rana, alongside with a heavily pigmented animal hemisphere as well as an unpigmented vegetal hemisphere. About 80% of the way into first cleavage, the cortical cytoplasm rotates 30 degrees relative to the internal cytoplasm. This rotation is important in that gastrulation will begin in the area which is opposed to that of sperm entry point. It is the region where the cytoplasm is being greatly displaced. See in frogs, gray crescent is localized adjacent to cortical rotation brings in the variety of pigment beneath the heavily pigmented cortical cytoplasm. Deep first cleavage furrow bisects the gray crescent. Gastrulation in amphibians. The amphibian gastrulation study serves as the oldest as well as one of the newest areas of experimental embryology. See Beechin 2001, Brockman as well as Gilbert 2005. Majority of theories regarding the gastrulation mechanisms as well as axis specification have been revised over the past 2 decades. The study of the developmental movements linked to gastrulation has also been complicated by the fact that there are no single ways amphibians gastrulate. Different species make use of a variety of ways to attain the same growth pattern. Currently, a lump sum of researchers is doing categorized research on xenophils laevis pronouncing the importance of gastrulation within that species. Amphibian blastulae are faced alongside with the same tasks as the invertebrate blastulae to bring inside the embryo those areas destined to form the endodermal organs to surround the embryo alongside with cells capable of forming the ectoderm, as well as to place the mesodermal cells in the proper positions linking the ectoderm as well as the endoderm. Unlike in sea urchin gastrulation, many of the cell as well as tissue movements in xenophos gastrulation happen simultaneously. We could, however, still identify the different types of behaviors that occur during xenophos gastrulation. White circle epiboly, the thin as well as movement of the animal cap cells in the vegetal hemispheres by way of proliferation as well as intercalation radially. White circle vegetal rotation. Here, the vegetal cells will be asymmetrically pressing up against the dorsal interblastoceal roof. White circle bottle cell formation and invagination. Apical constriction at the dorsal blastopore lip creates anisotropic forces that promote invagination. White circle cell migration and involution. The invaginating cells leading edge will crawl up onto the blastoceal roof. White circle convergence and extension. The targeted medial to lateral intercalation of midline cells. Convergence drives anterior posterior axis elongation extension. Figure 5.44 summary of experiments by Nukup as well as by Nakamura as well as Takasaki 1970 depicts that vegetal endoderm induces mesoderm. Animal cap cells when they are removed will form a ciliated group of ciliated epidermal cells. The vegetal cells when removed will form bud-like tissue. Further, equatorial marginal zone cells will generate mesoderm. B by combining the animal cap cells alongside with vegetal cap cells will trigger the animals to form the mesoderm. See model for sake of mesoderm induction in Xenophos. FGF2. Otherwise, the MP4 the ventral signals will be spread in the embryo's vegetal region. It will program the marginal cells such that it will form mesoderm. Whilst the dorsal side represents the region located away from the point of sperm entry point. In this region, a signal is being produced by the vegetal cells of the Nukup center. This signal produced dorsally will trigger the sperm organizer within the overlying marginal zone cells. Figure 5.55 organization of a secondary axis by dorsal blastopore lip tissue. A. The early gastrulous dorsal lip is being transplanted to one more embryo with the early gastrulous stage to a region defined to form the ventral epidermis. B. The donor tissue invaginates as well as forms a second archenteron, as well as then a second embryonic axis. Both donors as well as host tissues are seen in the new neural tube, notochord, as well as somites. C. As a consequence, one more embryo commonly known as second embryo will be generated that joins the pre-existing one. Deep structure of the dorsal blastopore lip region in an early xenophos gastrula. AC after Hamburger 1988, D after Winkelbauer as well as Scherfeld 1999 and Arndt as well as Nobler 1999. The dorsal signal, part 1, the Nukup center. Experiments by Peter Nukup as well as Osamu Nakamura show that the organizer receives its special properties from signals coming from the perspective endoderm beneath it. Nakamura as well as Takasaki 1970 illustrated the origin of mesoderm from the equatorial cells which are marginally placed within the border that links the animal as well as vegetal poles. Nakamura further brought to light that the key features of this fresh generated mesoderm can be induced by way of vegetal presumptive endoderm cells underlying them. Nukup then isolated the equatorial cells which makes presumptive mesoderm from that of blastula. He further highlighted that either the animal cap, i.e., presumptive ectoderm, or the vegetal cap, i.e., presumptive endoderm, will not form any mesodermal tissue. Whenever the two caps were undergoing recombination, the animal cap cells were triggered towards the formation of mesodermal structures, like muscles, notochord, blood cells, as well as kidney cells. The induction of polarity decides if the dorsal mesoderm is induced by animal cap cells else by ventral mesoderm. It actually relies if ventral endodermal fragment is from dorsal origin else ventral origin, while ventral plus lateral vegetal cells located adjacent to the sperm entry cycle induce ventral producing blood cells, mesenchyme, as well as intermediate kidney mesoderm. Dorsalmost vegetal cells specify dorsal mesoderm components like notochord and somites, inclusive of those with organizer properties. These dorsalmost vegetal cells which are able to induce the organizer are named as the Nukup center. The dorsal signal, part 2, beta-catenin. Once the special nature of the Nukup center was discovered, an important question became, what gives these dorsalmost vegetal cells their special properties? The beta-catenin serves as a major factor forming the Nukup center within the vegetal cells. Beta-catenin is responsible for specifying the micromers of the sea urchin embryo. Beta-catenin remains as the most important protein for inducing dorsal tissues of amphibian. Experimentally, if beta-catenin is depleted, the dorsal structures will not be formed. Also, if we inject beta-catenin into an embryo's ventral side, it will generate a secondary axis. In xenophos embryos, beta-catenin is initially synthesized throughout the embryo from maternal mRNA. Yost et al. 1996, Larabel et al. 1997. Beta-catenin remains to collect up in higher levels within the egg's dorsal region at times of cytoplasmic movements happening during fertilization. It also endures to accrue preferentially at the dorsal side throughout early cleavage. This accumulation is seen in the nuclei of the dorsal cells as well as appears to cover both the Nukup center as well as organizer regions. GSK3 is disarmed by virtue of GBP as well as disheveled. These binary proteins release GSK3 from the degradation complex besides preventing it from binding beta-catenin, targeting it for sake of destruction. During initial cell cycle, microtubules arrangement will be in analogous zones within the egg's vegetal portion. The GBP travels in conjunction with the microtubules. It adheres to kinesin, which in turn serves as an ACPase motor protein that travels on microtubules. Kinesin constantly travels towards the microtubule's growing end. It moves to the point which is reverse of sperm entry marking forthcoming dorsal region, figure 5.56. The shuttle, which is originally found in the vegetal pole cortex, grabs onto the GBP, as well as it too becomes translocated alongside the microtubular monorail, Miller et al. 1999, Weaver et al. 2003. Once at the site opposite the point of sperm entry, GBP as well as DSH are released from the microtubules. Here, they inactivate GSK3, allowing beta-catenin to accumulate as well as start the gene regulatory network for sake of dorsal cell specification while beta-catenin on the opposite side, future ventral face, is degraded, figure 5.57, Weaver as well as Kimmelman 2004. The regional specificity of mesoderm induction could be demonstrated by recombining cells of 32 cell xenophos embryos. Animal pole cells were labeled promoting easier identification of its descendants. The recombination experiments were carried out in inductions as consequence since these are concise below. D1, the dorsalmost vegetal blastomere, induces the animal pole cells, which forms dorsal mesoderm. Figure 5.56, the mesoderm's regional specificity of induction is well understood by recombining cells of 32 cell xenophos embryos. Figure 5.57, disheveled protein pacifies beta-catenin. A, in unfertilized egg, disheveled DSH associates alongside with a certain customary protein by the vegetal pole. B, upon fertilization, protein vesicles are dorsally translocated alongside subcortical microtubule paths. C, disheveled is then released from its vesicles and accumulates within the upcoming dorsal region. D, disheveled adheres and prohibits GSK3 action that avoids beta-catenin degradation on the embryo's dorsal side. E, the blastomere's dorsal nuclei will receive beta-catenin, whilst the ventral region nuclei do not. The TGF-beta family protein in the Nukup center could induce the cells in the dorsal marginal zone above them to express the transcription factor that would also bind to the promoter of the goosefoid gene as well as cooperate alongside with CMY to activate it. The contender for sake of this TGF-beta factor includes VG1, VECT, as well as nodal-related proteins. All these proteins are being produced within the endodermal region. Figure 5.58 instantaneous proceedings that are theorized to induce the organizer within the dorsal mesoderm are illustrated stepwise. Initially, cortical rotation triggers disheveled protein translocation towards the embryo's dorsal side. DSH will adhere to GSK3, letting beta-catenin to collect within the impending dorsal region. At times of cleavage, beta-catenin will make an entry towards the nuclei as well as adheres to TCF3. It internal code for a transcription factor activating the CMY protein. CMY and LIN1, a transcription factor triggered by TGF-beta pathway, will work with each other for activating the goosefoid gene. These goosefoids serve as the transcription factor activating proteins that are significant for organizers' activities. After Moon as well as Kimmelman 1998. When they repeated the Nukup animal recombination yet included a specific inhibitor of nodal-related proteins. Now, the vegetal cell's induction did not happen. While inhibitor exhibited no constraint on VECT, VG1, activin. Additionally, it was explored that at final blastula stages, three nodal-related proteins XNR1, XNR2, as well as XNR4 were expressed as dorsal ventral gradient within the endoderm. Such ramp is triggered by instigation of xenophos nodal-related gene expression. This in turn occurs by the synergistic action of VG1, VECT, and beta-catenin. AGS as well as his colleagues present a model in which the dorsally located beta-catenin as well as the vegetally located VECT as well as VG1 signals interact to create a gradient of nodal-related proteins XNR1, XNR2, and XNR4 transversely to the endoderm. A, stage 8B, stage 9C, stage 10 ventral dorsal. VECT, VGL. VECT, VGL. Beta-catenin. Beta-catenin. XNR gradient of BMP4XWNT8 ventral and lateral mesoderm. Organizer. Organizer. Nodal related high. Nodal related low. Ventral. Mesoderm. Figure 5.59 model for sake of mesoderm induction as well as induction of organizer by the interaction of beta-catenin as well as TGF-beta proteins. A. In final blastulus stages, vectin BG1 are localized within the vegetal hemisphere, whereas beta-catenin is spotted within the dorsal region. B. Beta-catenin has synergistic action alongside with vectin BEG1 for preparing the xenopus-nodal-related XNR genes. Subsequently, a XNR gradient is induced into the endoderm with higher concentration within the dorsal most region. C. Whilst the mesoderm is being specified by way of these XNR gradient proteins. Mesoderm having diminutive else total absence of XNR proteins will in turn have elevated BMP4, XWNT8 levels. Eventually they form the ventral mesoderm. Those with intermediate XNRs concentrations will form the lateral mesoderm. Elevated XNRs concentration will induce the expression of goosefoid genes that in turn activates dorsal mesodermal genes remarking the mesodermal tissue to play the role of the organizer. These nodal-related proteins specify the mesoderm such that those regions alongside with little otherwise no nodal-related protein remarks the ventral mesoderm. Regions with lower concentration of nodal protein will form lateral mesoderm. Areas containing higher concentrations of nodal proteins will in turn form the organizer. Subsequently, the expression of nodal-related proteins will trigger the expression of the goosefoid gene, while the nodal-related protein specific inhibitor prevents this activation. Functions of the organizer. The ability to self-differentiate into dorsal mesoderm, precordial plate, short mesoderm, etc. The ability for dorsalis in the adjacent mesoderm to form the paraxial semiforming mesoderm. Without the organizer, it would form ventral mesoderm. The ability to dorsalize the ectoderm as well as induce formation of the neural tube. The ability to initiate the movements of gastrulation. Nuclear protein secreted proteins X lim 1 shorten. X not DICOF OTX2 ADMPXFD1 FRZBXANF1 noggin. Goosefoid follistatin. HNF3 beta related proteins 4. Instance, Burkhead, Pintolobus. Sonic hedgehog. Cerberus. Nodal related proteins several. Table 5.5 proteins expressed solely otherwise almost exclusively in the organizer partial list. The diffusible proteins of the organizer I, the BMP inhibitors. Goosefoid protein is localized within the nuclear region. Subsequent to it, the genes that code for soluble proteins will take the function of organizer, which will in turn organize the dorsoventral as well as anterior-posterior axis. Primary indication of presence of diffusible signals from the notochord came from several sources. First, Hans Holtfrieder 1933 showed that if amphibian embryos are positioned within a high salt solution, the mesoderm will undergo vagination instead of invagination. It will not underlie the ectoderm. Such ectoderm is not able to form notochord and neural structures. In an experiment, researchers placed nuke dorsal tissue on one of the side of a filter, which was thin enough such that none of the process could actually fit over the pores. They placed the competent gastrule ectoderm on the other side. Subsequent to multiple hours of time observation, it was noticed that neural structures developed from the ectodermal tissue. The characteristics of factors which diffuse from that of the organizer took almost a century for identification. Current studies based on induction have resulted in a remarkable as well as non-obvious conclusion. The ectoderm is essentially triggered to form the epidermis. This is made feasible by way of presence of inducers like bone morphogenic proteins BMPs. The nervous system is being formed from ectodermal that is immune to epidermal induction. Hamadi Bragan Lund in 1997. Top findings. The ectoderm's default fate is to become neural. Certain parts of the embryo induce the ectoderm to become epidermal tissue, as well as organizer tissues act by secreting molecules that inhibit this induction, allowing the protected ectoderm to become neural. Noggin, shorten and nodal related 3. A cDNA plasmid library was constructed from dorsalis, lithium chloride treated, gastrally based on the experiments by Smith as well as Harlan 1992. RNAs were synthesized from these plasmid sets. These RNAs were given to the ventralist embryos that lacked neural tube. It was actually formed by subjecting the early embryos to ultraviolet radiation. The plasmid sets having RNAs redeveloped the dorsal axis in these embryos. Plasmid clones were retrieved from those containing mRNAs that were in turn able to restore the dorsal axis. One amongst them had the noggin gene. The freshly transcribed noggin mRNA will be initially localized within the dorsal blastoportal region, which subsequently is expressed within the notochord. When single cell were injected with noggin mRNA, those even if UV radiated will undergo complete rescue of the dorsal axis that permits the complete embryo formation. The noggin mRNA sequence indicates sturdily that it is a secreted protein. Smith in 1993 illustrated that noggin expression can execute two of the key roles of a feminine gold organizer. White circle noggin induces the dorsal ectoderm by formation of neural tissue. It also dorsalizes mesoderm cells. In absence of noggin, it will form the ventral mesoderm. White circle noggin binds to BMP4 as well as BMP2 as well as inhibits their binding to receptors. Shorten as well as nodal related 3. Shorten serves as secondary major organizer protein. It was indeed isolated from cDNA clones, from mRNAs localized within the dorsalis yet not within the ventralist embryos. Tests were conducted upon clones by way of injecting shorten to ventral blastomeres to assure that it can really induce the secondary axes. Clones with the capability of inducing a secondary neural tube will have the shorten gene being expressed. The shorten mRNA was limited to the dorsal blastoportal initially and subsequently it was trapped within the dorsal mesoderm of the notochord. Shorten will bind to BMP2 and BMP4 directly by preventing their complexing with their receptors like noggin. Within the zebrafish, shorten loss of function mutation will significantly reduce the formation of neural plate as well as the ventral mesoderm region. Hammerschmidt et al. 1996. XNR3 nodal related 3 serves as third organizer secreted protein synthesized by way of the organizer's superficial cells and has the capability to block BMP4. Follistatin. The follistatin serves as the fourth organizer secreted protein. It was located within the organizer by way of unforeseen outcome of experiment. Researchers made serendipitous invention when they were looking at the protein activin was key for mesodermal induction. The researchers constructed a dominant negative activin receptor which was later injected into xenopus embryos. Curiously, the embryo's ectoderm commenced to express the neural specific proteins. Activin receptor binds with further structurally analogous molecules like BMP bone morphogenic proteins which routinely bind with neuronation inhibitor. Whensoever follistatin was blocked, all of the ectoderm will form the neural structures. In 1994, Hamadi Bragan proposed the default model of neurulation. The organizer actually functions by fabricating inhibitors that blocks neurulation. Whenever the whole embryos elsewhere animal caps were dissociated, they will form neural tissue. This result can be reasonable if all the ectoderm does not default forms epidermis, yet forms neural to express an epidermal phenotype. Meanwhile, the obviously expressed follistatin will bind and inhibit activin alongside with other related proteins. It was theorized that it is secreted by the organizer. Using in situ hybridization by Hamadi Bragan as well as Melton 1994, it was concluded that the mRNA of follistatin is localized within the dorsal blastoportal as well as notochord. The BMP4 serves as an important molecule helping with epidermis induction. BMP4 acts antagonistic to the organizing molecules. BMP4 mRNA if injected inside eggs of xenopus, all of the embryo's mesoderm will transform into ventrolateral mesoderm. By contrast, if the BMP4 receptor dominant negative is overexpressed, it will lead to formation of dual dorsal axes. BMP4 triggers the formation of epidermis from ectoderm. Various labs establish that organizer secretes follistatin, shorten, as well as noggin which inhibits the binding of BMP to mesoderm and ectoderm localized adjacent to the organizer. Whenever BMP is bound with ectodermal cells, it stimulates the gene expression of MSX1, which in turn will trigger epidermal specific genes expression. It will inhibit genes generating neural phenotype. Within the mesoderm, BMP4 will trigger XFEN1, which is meant to form ventral mesoderm. Meager doses of BMP4 will induce the formation of muscles, while mediocre levels will make the cells to form kidney. Elevated doses will stimulate genes instructing the mesoderm towards forming blood cells. Organizers will eventually work by way of secreting BMP4 inhibitors whilst they do not directly trigger neurons. BMP4 aids as the active persuader of epidermis from ventral ectoderm, blood cells, as well as connective tissue from ventralism of the mesoderm. Likewise, the antagonistic organizers like noggin, shorten, as well as follistatin will inhibit its function. BMP4. Mesoderm. Ventral. Epidermal. Ectoderm dorsal. Dorsal. Endoderm vegetal. Neural. Ectoderm. Animal. Organizer molecules, shorten, noggin, follistatin. B. RELATI versus E. PROTEIN. CONCENTRATION. BMP4. Noggin. Ventrolateral A. BMP4 functions as the prevailing ventricular. Organizer proteins Jordan, noggin, with follistatin will prohibit the BMP4 expression. The opposed effects are noticed across entire trio germ layers. B. BMP4 elicits the patterning of the mesoderm by way of concentration reliant gene expression of a variety of genes. Marginal zone forming prospective mesoderm will exhibit BMP4 expression excluding the dorsal area. This is due to the fact that dorsal domain is dominated by noggin as well as Jordan. Routinely, organizers will bind with BMP4, which in turn prevents BMP4 to reach the mesodermal cells. The zones of noggin and Jordan expression will inhibit BMP4, thereby making them to form notochord, the organizer tissue. Zones that are a bit away from organizer will express MYF5, which serves as marker for dorsolateral muscles. By contrast, if elevated amount of BMP4 molecules are expressed, it would allow the expression of ventrolateral genes XFEN2 and ventral genes XFEN1 genes. After Dosh et al. 1997. The organizer 2 molecules, the WNT inhibitors. Endomesoderm will establish the dorsal blastophore lip. Existing studies highlighted that organizers function in two ways. One by inducing head structures, other way, they block WNT pathway by obstructing BMP4. Cerberus. XWNT, a potential gene of WNT member, helps in growth and differentiation. It represses the neural formation. XWNT8 undergoes complete expression all through the marginal mesoderm but not detected in the dorsal lip. Thereby, apart from BMP4, secondary antineuralizer was secreted by non-organizer mesoderm. Anterior-most head structures are induced by another secreted protein known as Cerberus. It is similar to all organizers. Cerberus aids the cement gland establishment, olfactory, nasal placoes, and eyes. Cerberus mRNA, if injected inside ventral side of the vegetable 32 cell stage xenoplasmia, ectopic head assemblies were triggered. The pharyngeal endomesoderm cells will express Cerberus gene that rise from early dorsal lip profound cells. Cerberus protein attaches to BMPs and XWNT8. FRZ band Dickoff. FRZ band Dickoff was produced within the endomesoderm which undergoes involution. FRZB, pronounced frisbee, is a tiny molecule, more like frizzle soluble form. Recollect, frizzle is a WNT receptor, which binds WNT proteins. FRZB is produced largely within the endomesodermal cells that were lying below the head. If there is too much production of FRZB, WNT signals will fade and embryos will not have ventral posterior structures, thereby they slowly become the head. The Dickoff German, big head, stubborn, protein undergoes extracellular interaction with WNT proteins. If we inject Dickoff antibodies inside the blastocele, it will make the subsequent embryos to be tiny, with deformations of heads but will not have any forebrain. B. Ventral animal. XWNT8. Vegetal. FRZB. Dickoff. Dorsal. WNT receptor, frizzle protein. A. XWNT8 FRZB. DSH. Figure 5.61 XWNT8 ventriculus is the mesoderm and inhibits the formation of anterior head within the ectoderm. A. FRZB anterior organizer protein binds with XWNT8 ahead of inducer binding expressor. FRZB is a soluble molecule that mimics the WNT binding domain of the WNT receptor, frizzle protein. B. XWNT8 is expressed throughout the marginal zone. The trunk structures induce BMP blockage of signals from the notochord. Yet, for generating a head, WNT signal and BMP signal must be blocked. Endomesoderm triggers blockage of notochordal signal and it forms anterior portion of the organizer. Figure 5.62 pathways differentiate ectoderm to form epidermis. With BMP signals, transcription factors of epidermis are being formed that activates the pathways which enables the cells for forming an epidermal keratinocyte. Wherever BMP signals are being lacked, transcription factors of neuralizing nature are being formed. Such factors in turn trigger neurogenin. Neurogenin transcription factor activates neurogene, which in turn differentiates cells into neurons. Figure 5.63 regional specificity of induction is well understood by implanting various regions of the archenteron roof inside premature tritura's gastrally. Such experimental embryos contain secondary dorsal structures. A. Head with balancers. B. Head with eyes, forebrain, as well as balancers. C. Posterior region of head and otic vesicles. D. Trunk tail segment after Mangold 1933. Figure 5.64 temporal specificity of induction. Mangold demonstrated that transplantation of early gastrolith to blastocellular early gastrolith leads to secondary axis while transplantation of late to early leads to tail. Early development in C. elegans. Caenorhabditis elegans is 1 mm long, free-living soil nematode. It partake embryogenesis within 16 hours, so it's welcomed amongst embryologists. Furthermore, the principal adult form serves as hermaphroditic, where organisms have the capability to produce both the eggs as well as sperm. Such roundworms reproduce by both self-fertilization and cross-fertilization with males. Figure 5.65 C. elegans. A. Sperms are being stored to enable the mature egg to move towards the vulva. B. The gonads. The germ cells close to the distal end will endure mitosis. Subsequently, meiosis occurs as it moves far to the distal tip. Primary meiosis leads to the production of sperm, which is stockpiled inside spermatheca. Eggs also undergo meiosis and in due course it undergoes fertilization as it crosses spermatheca. C. Initial development by way of movement of egg being fertilized and exits vulva. The germ cells arise from P lineage, stem cells. After Pines 1992, based on Sulston as well as Horvitz 1977 as well as Sulston et al. 1983 D. The germline undergoes 3 subsequent cell divisions to generate A, B, C, MS, as well as B lineages. A total of 959 somatic cells are present within the adult. After Pines 1992, based on Sulston as well as Horvitz 1977 as well as Sulston et al. 1983. The adult C. elegans has 959 somatic cells. The charm is that the entire lineage can be traced through by its feature of transparent cuticle. Also, contrast to cell lineages of vertebrates, C. elegans lineages are constant in all the individuals. Randomness is not at all noticed in any of the individuals. C. elegans genome is the foremost multicellular organism getting its genome completely sequenced with 19,000 genes. Cleavage and axis formation in C. elegans. The C. elegans zygote exhibits rotational holoblastic cleavage. In times of early cleavage, each asymmetrical division forms one founder cell, includes D, C, MS, AB, and E that forms differentiated descendants along with P1, P4 stem cell lineage. As a result of first cellular division, the cleavage furrow is asymmetrically positioned alongside the anterior-posterior axis. It is located adjacent to the posterior pole. It forms a founder cell AB as well as a stem cell P1. With second cellular division, the anterior founder cell AB will divide equatorially by way of longitudinal angle which is 90 degrees to that of the anterior-posterior axis. During the course of P1 cell division meridionally as well as transversely, it produces yet another cell, the M founder cell along with P2 the posterior stem cell. Further, the stem cell lineage will follow meridional division for generating an anterior founder cell, somatic cell, and a posterior cell stem cell lineage. The progenies of the founder cells can be visualized over the transparent cuticle which was term based on positions relative to their associated cells. For instance, ABL is the left hand daughter cell of the ABA cell, as well as ABA is the anterior daughter cell of the AB cell. Anterior posterior axis formation. The C. elegans extended axis will define the forthcoming anterior posterior axis of the nematode's body. The choice to find which of the determinants will form the anterior and which of them will form posterior in alignment will be decided with sperm from nucleus position. Whenever it arrives at cytoplasm of oocyte, the centriole associated with the sperm from nucleus will trigger cytoplasmic movements which will force the male from nucleus to reach the nearest end of the oblong oocyte. This end becomes the posterior pole. The secondary anterior posterior asymmetry is noticed just subsequent to fertilization, which triggers the migration of P granules. P granules specify the germ cells which are by nature act as ribonucleoprotein. When Strom et al. Used fluorescent antibodies against P granules, they came across interesting findings. Sooner after fertilization, the P granules that are haphazardly scattered will collect the posterior pole of the zygote. Eventually these P granules enter blastomere P1 that forms cytoplasm's posterior part. P1 cell having P granules is localized within the P1 cell of posterior region. Further, P1 granules transit from P1 to P2. So also, it moves granules to P3 and P4 subsequently. At end, P granules will be localized in P4 cell where the end products for sperm as well as eggs will be formed finally. But this gathering of P granules will require the presence of microfilaments. Surprisingly, they do not need microtubules. This was proved by treatment of zygote with cytochaslin D, a microfilament inhibitor that avoids the accumulation of P granules at posterior region. This proved that microfilaments are required for this process, whereas Dean-Colchine, a colchicine-like microtubule inhibitor, did not stop over the accumulation, indicating that microtubules are not mandate. The partitioning of the P granules as well as the alignment of the spindle fibers are together lacking in embryos of mothers which were lacking PAR partition defective genes. The proteins synthesized by PAR genes are localized within the embryo cortex and they intermingle with cytoskeleton containing the actin. Formation of the dorsal ventral and right left axis. The nematode's dorsal ventral axis is noticed during the AB cell division. In due course of division of AB cell, it actually elongates, in fact takes large size relative to eggshell. This triggers the cells to slide, that in turn localizes one AB daughter cell towards anterior as well as one to posterior making of ABA as well as ABP, respectively. This cuddling makes ABP cell to be placed above M cell. ABP is derived from P1 blastomere after its division. The ABP cell will outline the embryo's future dorsal side, whereas the M cell will serve as the ancestor of muscle as well as gut cells. It in fact symbolizes the forthcoming ventral surface of the embryo. At the 12-cell stage, the left-right axis is being specified. The MS blastomere is formed from the M cell division. It associates with part of the alpha cell, granddaughters, which distinguishes the right from the left. Control of blastomere identity. C. elegans validates both of the conditional as well as autonomous styles of cellular specification. Together these modes are noticed even supposing the initial two blastomeres were detached experimentally. The P1